same Yoruba people, but one lives in the rural environment, the other in the urban, which clearly shows the impact of environment on blood pressure. And the other cartoon there shows the sodium-potassium ratio. The average Nigerian, at least the one we studied, excretes about 100 milliequivalent of salt a day. In the Caribbean, it's about 121, and among African Americans, it's about 140. So that slope that you see in terms of blood pressure and hypertension rate, we were able to explain about 60 to 70% of it just by looking at how heavy you are, level of physical activity, and the level of salt consumption. That's the village where we do the work. And if you know something about Igbora has the highest twinning rate in the world. People believe it's in the yam, but I'm not sure. So I eat a lot of yam, and I actually have a twin, 2 boys. So since then, I've always appreciated the impact of environment on health, although I do a lot of genomic work. So your lifestyle, the level of urbanization. If you've been caught in traffic in Lagos, you see how you can get very sick. The tax policies, other educational policies, access to care are all as critically important as the gene that you carry in terms of your health issues. And in the United States, for example, I'm sure you heard about, the United States has the highest rate of incarceration in the world. And a lot of those people who are incarcerated in the United States are black men. So in my lab, we use a lot of, combine the gene and environment interactions to understand complex diseases, mostly metabolic disorders. So that's sort of the model that we typically follow. So another influence in my life is Georgia Dunstan, who I consider to be one of the most brilliant people I ever met in my life. She's a human geneticist. And so Georgia recruited me to Howard University after Loyola. And the story actually is a little interesting. Georgia was a fellow in Francis Collins' lab. The picture you see there with Francis is actually when he was a missionary in Nigeria after his training in medicine. And that's when Francis first noticed the rate of diabetes in this part of the world. But Georgia spent the time with Francis, and they both agreed that we needed to study diabetes and complex diseases. And I was invited to lead the effort to do the genetic epidemiology of type 2 diabetes. And that influence of those two individuals led to several important things in my life, including participation in the HAPMA 1000 genome, things to do with the African Society of Human Genetics, which I will talk a little bit about. So again, as you can see, the people you meet along the way, the people that help you to achieve certain things are critically important. And when you find people who care about you, hang on to them as much as you can, and be prepared when they offer you. So again, we also know about the sequencing of the human genome. I actually came into the picture after the human genome was sequenced in terms of the HapMap. So the human genome basically, told us the address of where things are along the chromosomes. But we wanted to know, how does this thing vary from population to population, from individual to individuals? So the international HapMap was set up to actually help us to identify the extent of and the scope of human genetic variation. And it turned out to be about 10 million SNPs on average along the genome. But we know it was not very practical to interrogate 10 million SNPs all the time. So there was a need to find a way to summarize that. Using the concept of haplotype and linkage disequilibrium, we were able to roughly estimate that about 500,000 tagged SNPs, which is basically saying if you have a chunk like this, and you're able to get one or two things, you can capture the whole information in that chunk, so you don't need to interrogate all of those SNPs. So that was one of the major achievements of HapMap, and it led directly to the development of GWAS arrays that we use today. And the first success story was the complement factor age in terms of age-related macular degeneration was a clear demonstration of the power of whole genome wide association study. So We engaged the Yoruba community in the Ibadan area and also the Luya and Maasai community in Kenya. I regret to say that my colleague that I work with in Kenya, Professor Duncan Ngere, passed away a few years ago, tragically. And this was a dedicated scientist, so dedicated that he was sick, but he actually came to attend the African Society of Human Genetics meeting in Cape Town, South Africa, and he never went back home. So I really would, I really do miss him. He was really a dear friend. One of the things that we also observed with the HapMap was that the arrays that were being designed, they were being designed based on information from European ancestry populations, and they display less efficiency when you try to use them to interrogate African genomes. So that was something that stayed with me for a very long time. And one of the ways that one can explain that is actually quite simple. African populations have more diversity. The human evolutionary experience has been, for the most part, on the African continent before some of us ventured out to different parts of the world about 100,000 years ago. That's why we share a lot of stuff. But those people that left and migrated to different parts of the world only carried a subset of the variation that existed on the African continent at that point. So, although we share a lot of things, I'll say that there are parts of the genome, even common parts of the genome, that you can only interrogate on this continent because they just did not leave when people migrated out. So that is very, very important. And human genetics stands to gain, I say, immensely from the systematic sampling of African people. So the way I also try to explain this, and I know people like Francis and are probably tired of hearing it from me, is that if science was truly a logical process and a rational process, if you were going to sequence the genome of one person, you should come to Africa and throw a dart. Anybody you pick probably will give you more information than any other parts of the world. But that was not what happened. Again, part of the issue being who was putting up the money and where the scientists were at the point. So this is just another cartoon from the 1000 Genome to show that African ancestry populations have the highest level of variable size across the genome. Both things that are functional and not functional, at least our knowledge today, are all represented in that context. And hence, if you take a Yoruba person and a Maasai and compare them, they are more likely to be different from each other than when you take one of them and compare them to a Scot person. So that to me is probably the easiest way I communicate the level of diversity that exists on the African continent and the reason why we need to systematically study it. This is another way to describe that. I think 2010, myself and Lynn George, a very good friend, we wrote this paper where we basically were trying to characterize and describe how to interpret human genetic variation. One of the examples that we gave was hypersensitivity to abacavir treatment for HIV based on this HLA variant. Among the Maasai, probably the highest rate in the world is about 14%, and we didn't see it among the Yorubas. And therefore, if you describe that in the context of being an African variant or African people or black people, you completely distort the distribution of this variant, and it will lead you to a wrong public health decision as to who to screen. Again, that is why we need to study multiple populations across the African continent. And as part of the H3 Africa, which I will talk a little more about, we have now designed, working with Illumina, have now designed a more efficient array, which is the yellow bar there, to interrogate African populations. So I encourage you, for those who are going to do genome-wide association, to look into this chip, in terms of the work that you want to do. It will be more efficient, and you will be able to capture more of the variation that exists on the continent. Sorry. Oh, what did I do? Okay. All right. So the other thing that happened is that after about 10 years at Howard University, myself and Francis Collins, I told him that I was ready to move. because I think it got to a point where I felt like the environment will not be able to give me the necessary things I need to go to the next level. I also wanted to make this point to the people, young men and women in the audience, to say that knowing when to move and knowing when to stay from your mentor to the next level is critically important in science, because if you do it at the wrong time, you may compromise yourself, and if you don't do it, you may also compromise yourself because you are not able to stand out as an independent scientist. So one of the things that happened is that I brainstormed with Francis Collins and Eric Green. Actually, at that time, Francis was not the director of NIH. He was the director of the Genome Institute, and Eric Green was the scientific director of the Genome Institute. But both of them have risen over time. And we brainstormed to establish this center for genomics and global health on the NIH campus. So again, another influence. And again, just to let you know a little bit about the NIH, it's probably the largest biomedical research institution in the world. And for me, I always, every now and then, I pinch myself. I say, Charles, you grew up from Upper Lawani in Benin City, and now you're working at the NIH. Sometimes I don't believe it, but it is true. And the level of science that goes on this campus is absolutely fabulous. One of the things that the NIH has done for me personally is that it's always good to realize that there are so many people who know so much than you, and it makes you humble, but more importantly, it gives you avenue of people's brain to pick when you have questions, difficult questions. That was the NIH campus has done for me. A lot of scientists, practically about every question you're asking about biomedical research, probably going on that campus in a sense. So it has been extremely helpful for me. One of the motivation for the work that I do today and the creation of H3 Africa was the realization that after the HapMap and a spin-off called ENCODE that was helping us to understand the function of the variations that we are identifying, that out of the 32 labs that were participating in this international effort, none was in Africa. To me, that was absolutely depressing. In a way, so I, was just saying we need to do something different. So another way to express that, these are recent sentiments from some of the press that have come to interview me and all of that stuff. One of the things that is really important to me is that we engage the global population, especially African population, in genomic work. That is just critical to me, and it has led to a very vocal push to make sure that we ensure that tomorrow's medicine will work for all, not just those who can afford to do genomic work. And diversity in genomics continue to be a major, major problem. Programs like H3 Africa and Delta program are going to begin to address that. The issue of diversity in genomics is not only a social justice issue, it's a scientific imperative. You cannot, like I said earlier, there are certain parts of the genome, a certain part of human story you cannot tell without engaging the global population. An example is this press release and study that was done and published in the England Journal of Medicine, that black people in America, African American, were actually being misdiagnosed as having heart condition because they were being told that they carry pathogenic variants that put them at risk. But it turned out that it was completely wrong, because the reason is that the reference that were being used did not have enough variation from African people, and therefore the wrong decision was being used. The other thing is that discovery can be compromised. The PCSK9 in treatment for LDL cholesterol was discovered in African Americans because it's more common, but it's actually applicable to global populations today in terms of treatment. So if you don't engage different populations, you can compromise science, apart from the social justice issue associated with that. So all of these sentiments led us to the creation of the African Society of Human Genetics, and the current president is here, Ambrose. I served as the founding president for over 10 years, and I tell you, it's a vibrant society, and I will encourage everybody to please become a member, and I make that advertisement without apologies. Really, really become, because it has been instrumental in establishing the H3 Africa program, which I want to talk about right now. And I put this quote here from Shakespeare to basically say, when we were thinking about establishing something like H3 Africa, we had no clue where the money was going to come from. We had no clue who was going to listen to us. And but we kept pushing. We kept pushing. And we're hoping that somebody will listen someday. And Francis Collins, again, listen. This was a meeting we had in Cairo. One of the good things about the African Society of Human Genetics is that we go to different African countries to have our annual meeting. And the next meeting is going to be in Mali. We're, again, extremely excited about that. So Francis was here and helped us to brainstorm about what we can do. At that point, we were calling it the African Genome Project, but it transitioned to what we call H3 Africa today. And the motivation, again, is what is written there. Basically, we want to make sure that the revolution of genomics doesn't, again, go past Africa, and that we have the necessary capacity to drive that. And so we got the attention of the NIH and the Wellcome Trust, and it was launched, again, in 2010 at the headquarters of the Wellcome Trust in London. And the individuals you see there, starting from the, I think my left, so is Bongani Mayosi, who was the Dean of Medicine in Cape Town, also a very, very dear friend who unfortunately also passed away. And we really do miss him quite a bit. And Mark Wolpert, who was the leader of the Wellcome Trust at that time, Eric Green and Francis Collins, and this crazy man standing here before you. Again, it was actually quite interesting kind of meeting, and we developed what we now call H3 Africa with this vision to enhance the capacity for using contemporary research approaches in Africa by African scientists to understand the genetic and environmental factors that determine disease susceptibility and drug response in Africa. The last bullet there in red was perhaps the most critical aspect of this. We wanted to establish a resource and a funding mechanism that the money goes directly to the African investigators in the African institution so that they are the one driving the bus, so to say. You know, the person that has the money tend to dictate what happens. So H3 Africa has been remarkably successful. The initial commitment was $38 million, and the first phase went up to about $76 million, funding about 25 research projects, 27 African countries, over 500 investigators across the continent. And it has now grown. We are now in the last 5 years of funding, at least from the Common Fund perspective, to about $170 million of funding now, 48 projects across different African countries, with the goal of enrolling over 80,000 Africans, so we can begin to have the necessary sample size to actually do things like genome-wide association studies across the continent. And lots of trainees have gone through this program, and multiple publications are actually coming out. And again, I'm extremely, extremely proud of what H3 Africa has done across the country. Before H3 Africa, I like to put this cartoon to show that African scientists, they were doing extremely good work, but they were not talking to each other. Why? They were basically following the money. They talked to people in the UK, talked to people in America, talked to people in Asia, but you hardly find large collaborations between Africans across African countries. But H3 Africa has indeed changed that dramatically, where people now are collaborating across national boundaries and across institutions and driving science in a very, very big way. Again, congratulations to H3 Africa. So I want to give you a little bit of science from H3 Africa, and then I'll tell you about what we just discovered in my lab and end with that. Again, part of the thing about H3 Africa is really trying to put together the necessary genomic resources so that African scientists can start telling the stories about genomics on the continent. So this paper is under review in Nature. We just got the initial feedback from Nature. We are glad that it wasn't rejected, so there will be an opportunity to respond. The young men and women who are driving that are actually working real hard at this point to try to understand that. So a little bit of sneak peek into what we found from doing this kind of work is really trying to understand the story that has been told before in terms of migrations across the African continent. One of the things that I really want to highlight here is that red circle is the Boron population from Nigeria. It's the middle part of Nigeria. For whatever reason, we don't know how and why, they carry quite a bit, up to about 14% of Nilo-Saharan genetic background. Now think about that for a minute. There's a genetic background that you see in places like Kenya, Uganda, but these people in central Nigeria carry quite a significant proportion of Nilo-Saharan genetic background. We don't know where it came from, how it got there. So it's a very interesting story, and that is why I keep saying the story of Africa, at least in terms of migration and genetics, we are just beginning to peel it. We are just beginning to peel it. So whatever you're reading now, read it with a fat grain of salt. We are, again, beginning to tell some of those stories. And the other outstanding thing is the high proportion of East African mitochondrial haplogroup that we see in this group in central Nigeria. That was absolutely, to me, fascinating part of the human genome. And one way I look at the genome actually is a history book. It's a history book that has unbiasedly captured human experiences over time. And we are just learning to read it, to read that history book. And it's going to tell us some very superb stories. So one of the things that has been done in places like America and in Europe is to identify medically relevant variants so we can tell people when we find them. One of the things that we found in this study is that at least 17 individuals in the African community and samples that we use are actually running around and are quite healthy, but they carry this gene that has been classified, or this variant, as pathogenic. So what is going on? So we need to understand all of this. Pathogenicity is really, may have to be, we have to rethink that once we are able to study more African populations. And the other story here is the relationship between groups that are very, very close together, but have very different history in terms of their history and also in terms of medicine. These are groups in Uganda, Bantu group and Nilo-Saharan group, and they have very, very different migrational background and also different susceptibility in terms of malaria. So very fascinating story that we're just beginning to, again, appreciate. And these are the young men and women and also young men and women that did the work, and I'm extremely proud that over 90% of the people who did this work that is under review in Nature are African scientists in African institutions. So I am absolutely thrilled by this effort. Now I want to tell you a little bit about genome-wide association study and how we have used it in our type 2 diabetes. I put this here just in case some people have not, don't know much about genome-wide association or have not done it. Basically, it's a tool that we use in genetics to interrogate, where a signal may be in the genome. Basically, is really based on the fact that we don't know, and it's a very agnostic process. So we basically line the genome up with markers, and we look for where we find signals. So where we find association between cases and controls, that is basically the research design. As I said in the HapMap, these variants that we put on this GWAS chip are not necessarily the causal variants, but we are hoping that they track the causal variants. So when we do more fine mapping and interrogation, that we can find it. Over 400,000 genome-wide association study has been conducted to date. And for diabetes, it's been absolutely successful tool to use with important variants in almost all chromosomes for type 2 diabetes. And over 243 loci have indeed been identified for type 2 diabetes. But the problem is that most of these have been done in European ancestry population. This is the first comprehensive, large-scale study of type 2 diabetes using an African efficient GWAS array for our study called ADAM in Nigeria, Ghana, and Kenya. So we have cases and controls, about 2,500 each or so, which we carefully characterized. Again, when this study was reviewed initially, the reviewers were extremely skeptical whether you can do this kind of work in the African community. And, we were able to convince them, and they don't ask that question anymore, anyway, in a sense. So we've done the work, and what we found. Again, this cartoon is called a Manhattan plot, and the reason is that when you're in Manhattan, in New York, you see these high rises. So when we see this signal that pops up, we have this plot called Manhattan. So each of those big things there is representing a high rise of an association between a set of variants and and the phenotype that we are studying. I want to highlight ZRMB3. That variant, despite the fact that we studied close to 1 million European ancestry individuals in GWAS in the context of type 2 diabetes, this particular variant in gene has not been picked up. So we discovered this in our diabetes project in West Africa and East Africa, and we were able to replicate it with Aisha in South Africa for the Zulu sample that she had. Okay, so initially, we did about bioinformatics assessment, and because we didn't know what this gene does, we just did not have any information. And we were very pleased to see that using existing expression databases that ZRMB3 is actually expressed in tissues and organs that are relevant to type 2 diabetes. So that was our first initial excitement. And then, as Martin said yesterday, science is a collaboration. It's a big enterprise, especially genomic work. And once we discovered that this gene is expressed in relevant tissue, we wanted to do more functional assays because we believe that's the only way we can get them into a reasonably high journal. So we collaborated with Noran's lab at University of Maryland. She's a zebrafish expert in the context of metabolic disorders. So she did some very fancy, interesting work, and perhaps more importantly, she was extremely patient because one of the reviewers drove us completely nuts. It took us about 18 months for us to respond to reviewers' comment. It took us 2.5 years to do the study, but 18 months to respond to reviewers' comment once it started, and she was patient all through. So using CRISPR-Cas9, she did several things. So we were able to identify the mutation in F2 generation of the zebrafish using, again, CRISPR-Cas9, in transgenic models and also in wild type, and in both genomic DNA deletion in exon 4 resulted in a frame shift and a stop codon that is displayed there in this sequence information. And we started to say, where is this thing expressed and how is it expressed? So we did RNA-seq, and we were very happy that we were able to see expression of this of ZRMB3, and I will replicate that expression using qPCR. But the real story is here. When we suppress this gene or we knock it out using sRNA or just using CRISPR to remove it, you see a significant drop in beta cells. So you see the controls and the mutant having much more reduced beta cells, and the right cartoon there is the just the count, and also shows that there's much reduced beta cell number. So this is when we knew that we are really up to something here. And then we show also reduced expression of ZRAMB3, again in the mammalian cell line. I'm not sure what happened. Okay, mine went out. And then this was a definite experiment in the sense that the cells that we silenced or downregulated this particular gene expression did not respond to very well in high glucose environment. So the production of insulin was seriously compromised, which is consistent with the reduction of beta cell mass. So we, again, this is when all three reviewers said, okay, we accept your paper. And I'd like to end by telling you my lab is a continuous revolving door. On average, I have 20 people in my lab, and they come from different parts of the world. And one of the things that we do is that occasionally we'll have what we call a potluck, where people bring food from different parts of the world, their cultural food, and it's absolutely wonderful. So you hear people from China, people from different African countries, and South America, and you see that lady. I'm not sure if my pointer will do a good job, but she's Caroline is from the Netherlands, and I don't know how I always manage to find myself close to her because she makes me look so short, so I really appreciate this multicultural. We call our group a United Nation in the lab, and that is how I try to train and encourage different people. And that's me there trying to be a warrior that I know nothing about, but that was a picture taken with some very interesting guys in Zambia during one of the African H3 Africa enterprise. So I thank you very much, and I'll take any questions that you may have.
Thank you very much. Excellent presentation covering everything from burgers to genetics to zebrafish. I must say, I relate very well to the burgers issue. When I first got to the US, I was also quite stunned by the amount of bread I was being fed, all the time. So that is, I guess 50 years on, it's still the same story. Yeah. All right. So I believe we have about 10 minutes for questions, comments. Okay, I see hands up there. So as we've been doing, we'll take 3 and then 3, and then we'll see how much time we have. Please just mention your name and your institution, and then your comment or question. Thank you.
Harriet Manja from Makerere University. Thank you for that talk. Sometimes talking about genetics in Africa by African scientists is like talking p-value to politicians or neuroanatomy to lawyers. Our IRB, when they hear about storing samples for genetics, they put on the brakes, they jam, because they say there are risks, and none of us are sure what the risks are. And you talk about H3 Africa, we've heard a lot about it, but if you ask many young scientists what it's all about, they say they're storing samples for something. So my question, isn't it time now to start training us in Africa a little more about genetics beyond what we learned in physiology? Isn't it time to start training our public and more people about genetics? Isn't it time to start having meetings in Africa on genetics? So we train the next researchers who will then do the studies in Africa rather than storing samples, convincing IRBs to ship them to the US for the analysis. So I think we need more activity on genetics in Africa so that we can answer the questions you posed. But thank you. Great presentation.
Again,
maybe we can, are there any?
So again, basically, the sentiment you express is the reason for H3 Africa. And we are indeed doing all of that now on the African continent. Even the biorepository where the samples have been stored for future use is on the African continent. The bioinformatics infrastructure is across different African countries. So one of the things that we really emphasize with H3 Africa is the issue of capacity building, to make sure that the analysis and all that, is being done locally. So the issue of IRB, I think one of the success story of H3 Africa really is that when we first started, and we were talking to IRB about broad consent so that genomic data can be shared globally and within the African countries, IRB were very skeptical, understandably so. But since then, we have actually worked together to develop a framework, again, with the help of the African Academy of Sciences, that is now published. So you can look at that framework in your own area, and if your IRB is not aware of some of those findings and documentations, I would seriously encourage you to look on the H3 Africa website. It's h3africa.org, and also a publication from the African Academy of Sciences that has put all of this together in a way that should make it much more friendly to do this kind of work and get them approved by local IRBs.
Any more? Yes, Tom.
Charles, thank you very much, and really want to thank you for accepting our invitation to come out and speak to this group. So in thinking about the future of H3 Africa, one of the things that have struck me is the fact that we're still talking about fairly small sample sizes in terms of the cohort sizes. And I wanted to hear your perspectives about the need for us to start thinking about sort of the big, ambitious approaches to building large population cohort studies that would really allow us to get into some of the things you are discussing today. Because, when I look at the level of participants for something like the UK Biobank or the China Kaduri Bank or all of the US initiative you are involved in, I think what we have in Africa pales into comparison with those. And I just want to hear your perspectives about what would you think would be the next phase of development in terms of large populations, cohort studies, and incorporating genomic studies?
Yeah, that's an excellent, again, question. I fully appreciate that, especially with somebody who does some of this genomic work. And because of that comment you just made, our last meeting that we had in Egypt, in Cairo, we actually proposed that the next phase from H3 Africa should be a 5 million cohort of individuals across the African continent, so that we can begin to have the number of engagement of African communities and also individuals that will sufficiently statistical power to interrogate some of this variation that we're interested in. One of the irony of the sort of the architecture of the African genomes is that we have this more variability and things are more fragmented. Therefore, you actually need a much higher sample size than you would in European population, because you have a segment like this in European population and African population, that segment is probably broken down to 20 parts. And therefore, for discovery, you actually need most individuals, but for fine mapping, the African genome is extremely efficient, because to narrow things down, because of that history of fragmentation. So, Tom, you are absolutely right. We need to start thinking even beyond sort of the 80,000, 100,000 that we now have in terms of H3 Africa. Absolutely.
There's a question at the back. I think whilst we are moving the mic to her, I recently was part of a meeting on another initiative called the Human Cell Atlas project. I don't know if you've heard about this, and what are your thoughts, and what are the things we need to look out for? I think they are probably starting off like where all these other projects started, and then they are now taking steps to bring in African representation. So what are your thoughts about that?
Again, one of the things that I guess global scientific community is fully aware now is that the imbalance that exists in all of these efforts. And again, we can't blame people too much. As Tom will testify to, is that we have been relatively very unsuccessful in engaging our governments in Africa and also private funding individuals to invest in this kind of science. So it's almost like the African engagement in all of these activities sort of come later. That seems to be the consistent story here. It's not that scientists don't understand the utility and how important it is, but it's just the way money is, where the science is, and all of that stuff. So I would say, go ahead to be absolutely participate in it and help shape it so that Africa, is really well represented in that effort. Yeah.
Thank you.
Yeah, I have the mic. So thank you very much. It was a very interesting talk, and it's just very exciting to see all the potential there is of bringing or putting Africans at the center of genomic research. My question relates to the engagement of Africans, just because from a lot of what I've seen in genomic medicine is the fact that it's one of the areas in science where it's very, very important to kind of really engage very well, not just with the scientists, but with the public, just because there's a lot of conflation of what it actually means, and because of historical reasons, just the idea of conflation, for example, with race-based medicine, and what it actually implies to be able to do science and try to differentiate people. Just to hear, you didn't say much about that, but how much effort is H3 Africa putting on real public engagement with the public to really have a real conversation in a way that the public can shape what is being done and not just scientists taking samples?
Yeah, again, the H3 Africa is really sees your comment as an important thing, and therefore has made some very important strides in that regard to try to be very engaging. So community engagement in all of these projects that is under H3 Africa, wherever possible, is highly encouraged to have this engagement. And one of the things that I also have done, if you look at something like the HapMap, for example, when we did HapMap, we had some very, very detailed community engagement. Perhaps from our own experience, the best so far that I have been involved in. And HapMap was done many, many, many years ago. We still send information to the communities in Kenya and in Nigeria about how the HapMap samples are used and what are they being used for. So those communities still receive letters, and they know how those resources are being used and how their effort has actually moved global effort to understand human history and health is going on. So I think it's really something that we have to do. Other things that H3 Africa has done, I think a cartoon series have been created to help the lay people to understand genetics. So that is also available. And so there are various aspects of this that is going on under H3 Africa, because again, we believe it's critically important, especially when we come to the level of translation of the genomic findings, again. So.
All right, two more questions, one over there, and then Professor Tobias is here. If the microphone can come here also.
Okay, thank you very much, Professor. I'm Esokum Kevin, Belgium master student in Kenya, and I'm very pleased with your presentation, and your story is particularly motivating. And I'm a very passionate student in human genetics. I'm not getting any formal human genetics training yet, but I'm very passionate about that. And my question is more directed. We know that the genetic diversity in Africa is really, really huge, and from your presentation, you highlighted some points. You showed some figures that showed some Africans and European population, and he talked about this genetic diversity. And I just wish to ask that, do we need to revisit the way we classify our local DIMs, our local subpopulations in Africa, considering the fact that these populations are very diverse? For example, if you take the Yorubans of Nigeria, the Essans, and the other ethnicity that you just showed, that you've discovered some genomic island in them, like that's the Nilo sub-Saharan genomic region. And also, like you consider the Gambian Mandinka, the Wolof, the Jalo, which are also very genetically diverse. And given the fact that our genome accession study in Africa, which is performing very poorly due to this high genetic diversity, considers all of these ethnicities as a single population, do we really need to revisit the fact that these ethnicities are very different and maybe consider these ethnicities as separate populations and perform genome-wide association study on separate ethnicities, and then probably pull these ethnicities together into meta-analysis, like what is being done in Europe or in other African populations? Because classically what they do is they just put together African populations together into meta-analysis. Thank you, sir.
Yeah, again, thanks for that question. I'll answer your question from that study that I described, where we found a new gene for type 2 diabetes. As I indicated, there are populations from Ghana, Akan, and mostly Ga, and Yoruba and Igbo from Nigeria. And in the Kenya environment around Eldoret, we had multiple ethnic groups that participated in that study. Now, for me, African national boundaries are extremely problematic when it comes to genetics. Most of those boundaries are colonial boundaries, and they have no bearing on the relationship between ancestries and the relationship between ethnic groups. It just doesn't make any sense. You have Yorubas in Nigeria, you have Yorubas in Benin. You know, the same group, but there's a boundary there that is national boundary. But in genetics, those things are completely uninformative, those boundaries. So my advice is that we usually will ask people, where is the ancestry of your father, where is the ancestry of your mother, where is the ancestry of your grandparents, and all that, and we take all of those into consideration. But perhaps more importantly, I don't think I showed you in the interest of time, we always do population stratification adjustments, and in all of our GWASs, and that is basically we tell the computer to cluster people based on genetic information, irrespective of what they've told us about ancestry or ethnicity. That way, we use those clusters to adjust our analysis so that the association that we are finding is not driven by heterogeneity due to ancestry, but it's actually due to association with the trait. You know, so that is really, so your point is worth taking. And when we do this kind of work, we really need to allow our computer program to tell us the stratification that exists in our sample and try to adjust for that stratification by doing principal component analysis.
Thank you. Last question.
Thank you. It's not a question per se, it's a comment. I think your story is quite uplifting to many African scientists, and it's an honor for me to be here to listen to what you've done over the years. My name is Tobias Chirwa. I'm the program director for SACAB, which is a statistical consortium. And from what you've said, I think we can see that there's a lot of gap in terms of the statistical analysis required within the African setting per se, and speaks to the need for capacity building within that particular setting. So I think we need to engage more, and it's something that I've been engaging with the Sydney Brenner Institute of Molecular Biology in terms of the gaps that we see for statistical analysis. But what I wanted to really seek your comment on is you mentioned that it's important to know when to move and when to stay. And I think it's a common problem in terms of diaspora, or even just within the African setting, people moving between countries. But I think it's a movement which I think is beneficial for the African region. But I want to hear your comment in terms of how you look at those particular movements, because there's a lot of people feel guilty moving out of their own country to another country, and yet the contribution is for the African region. So I just want to hear your comment on that.
The way I look at it is, I'm an African. Nigeria happens to be located within that structure. And wherever I can do my work on the African continent, I will go there. That's just me. And so my answer to your question is, I don't think we should be so hesitant if the opportunity to do good work exists in Ghana instead of Nigeria. Why should I hesitate to go to Ghana, to get that work done? In the end, if we do this successfully, we will lift up Africa. And all the other nations that are not currently participating will begin to also benefit from it. I think I seriously encourage everybody here to look at this continent as what we need to make better. And if it just happened that you are doing that within your country, fantastic. Ambrose is from Cameroon. He says he's genetically from Cameroon and professionally from South Africa in Cape Town. He's doing a fantastic job. Perhaps he couldn't have done such work in Cameroon. But the work that he's doing in South Africa has serious implications for multiple African countries and a global population. So I don't think we should hesitate to do that. I know it's a little bit more difficult to move, but if you have the opportunity, please take full advantage of it. Regarding infrastructure for statisticians and bioinformaticians, I can assure you that's a global problem. I don't think there's an unemployed bioinformatician in America. It's such high demand, so if you have that kind of skill, I would seriously encourage you to just get better, and you will be in high demand for statistical genetics, for bioinformatician, and we need to invest more in that regard. And that's why I'm also very proud of the H3 Africa BioNet that has over 300 people being trained in bioinformatics across the African continent.
All right, so I don't think there could have been a better note to end this, which is to say that we are together. We are all in this together. It doesn't matter where you are pushing, we have to push in the same direction. And we have to look out for each other and support each other. And, we would all get there together. So I want to thank Charles very much. It has been very, very, very interesting. And I don't know how many factorial Alphonse would sanction for Charles. Whilst we are doing the factorial, we'd like to invite Professor Nelson Toto to make a presentation to Charles. So, Alphonse, how many factorials are we going to do? Where's Alphonse?
I have the honor to ask the chair to make that decision.
Okay, yesterday we did 5. Since today is better, we do 6. So let's do the 6 factorial whilst Nelson's coming up. So. All right, good. Thank you very much.
Extremely appreciative of that. Thank you very much.
Is that a loudspeaker?
Okay. Wow, that's been an excellent session. And before we break for tea and poster viewing, about 2 announcements. The first one is that you should not take the headsets outside this venue. The second one is that for those who have airport transfers or who are traveling today, that you visit the AAS desk to finalize on your arrangements. However, if you've enjoyed Dakar so much that you want to stay and you miss your transport to the airport, then the other costs are on you as an individual. So let's break for coffee and tea and poster viewing, and then we come back at 11:30. Thank you very much. You can clap for me. I can see you are not in a hurry to go for
achieving results, and then they can spend that money on the research enterprise as well. I think one of the things that we've learned in the last 5 years, and it's been implemented in the new phasing of this project, is the need for not just centers of excellence in terms of research, but also the institutional impact and the institutional structures that support the research environment. And this, I think, is a key area in terms of leadership support, but also the research management capacity of the institutions as a whole. And the bank, of course, is a funder, and it does not have a huge capacity in terms of research management internally. So from our point of view, it's essential to build the capacity at institutions, but also to partner with the experts in the field to provide technical assistance to enable that capacity to be built at the institution. So I think that's the real focus in terms of what we're looking at beyond just funding research and postgraduate education, is to ensure that we bring in and work with the appropriate partners to build up the capacity on research management at institutions.
Thank you, Graham. So moving on to just say, Graham has just talked about capacity development at institutions and what is referred to as institutional impact. Do tell us your experience in trying to build research management capacity at institutions.
Thank you very much, Alan. And I must say thank you again for inviting me to be on this panel. But also, I think perhaps most rewarding for me is that research management has now become almost center to the work of research institutions. We have obviously, I've been in this field for more than a decade, and it is rewarding to see that it has come front and center again. It has had its ups and downs, but recognizing the importance of it, and at this meeting is very critical. So what have I been doing? I must say I'm wearing two hats here. I'm the co-director of the Alliance for African Partnership at Michigan State University in the US, but I'm also speaking from my experience as the past president of the Southern African Research and Innovation Management Association. I was the president from 2015 to 2017, a member for a very long time. We will be celebrating 20 years of existence in 2020. And so you are welcome to come and join the celebrations that we will be having next year. In terms of what we have been doing both at MSU and also at SARIMA, it's perhaps several things. We've always had, obviously, a capacity building portfolio and a capacity building initiative across Southern Africa. And our focus has typically been for the individual development of research managers. We've, over the years, been having workshops in different regions at our annual meetings, etc. But within the last 5 years, I would say, we have taken on professionalization of research management as a very key initiative. We've seen that this is an area where research managers have asked for a career path. They have asked that this become a discipline which can be taught at universities. And this is already being done in the United States, the UK, and many other institutions. So in African institutions, we are playing catch up, but in a very quick way. So what have we done so far? We have sort of had a needs assessment regionally within the Southern African region, but also incorporating the feedback from our colleagues in West, East, and Central Africa. We've developed a professional competency framework. We have looked at a career path for our research managers, starting with persons who are at the administrative level all the way to directors and and DVCs of research. We now have clearly laid out competencies that research managers should have. we have developed a professional certificate in research management for people who are entry level in the research management space. And this is an online program that is available at Wits University in South Africa. We're also working through an EU program to develop a postgraduate diploma in research management, and also developing the curriculum for an executive master's in research management. Those are on the table, and one is the professional certificate has begun. It was piloted last year with the Science Granting Council's initiative, and I think we will be opening this now to the general public of research managers across the continent this year, later on in this year. We also have one other program that is based on prior experience. So we recognize that there are those who will come, wanting to go through, if you're entry level, you take this, you take the certificate, you take the postgraduate diploma, et cetera, but then there are those who have been working in the field for many years, and so we recognize this experience, and we now have as well a professional recognition program in research management, and you can get it at various levels. I think I'll stop here.
Thank you, Jose, for that elaborate description of the work you're doing to really professionalize research management on the continent. But I'll move on to Sam. I know you have built and set up a very strong capacity building program for scientists. Whilst the efforts to build capacity for the research managers to support these scientists is still underway, could you describe to us how you do support your scientists and help them grow through the ranks?
Yeah, so thank you very much. And again, just to echo what has already been said, that the growth of a scientist is more than just the technical training that they receive from their supervisors, and that the growth is dependent on the environment within which they are growing beyond just the supervision. And for us, I would say among the key things that we use to support our scientists in growing is ensuring that they spend most of their time focusing on doing the science and not doing all the other extraneous things. And that means having a setup or a structure that provides very strong operational support. So it's not a scientist who's having to chase up and down on procuring simple things or organizing their travels or things like this. And this may look like minor things, but I am aware that in many institutions, this end up taking up a lot of time of the scientist. So that is at one level. The next level is ensuring that the infrastructures within which they're working are well managed and well supported. So, whether it is the physical infrastructure or whether it is managing their grants for them, whether it is ensuring that they are meeting the funders' conditions. And so together, what we're doing is creating an environment that allows them to focus primarily on doing the science, whereas we're providing all the operational and all the regulatory issues. And I maybe give an example for our clinical trialists. What we realize is that with large clinical trials, it's possible for the trial to employ everybody that they need within that trial, but often you'll have small clinical trials that cannot actually manage to employ all the people, support people that you require. And so we put together a clinical trials facility whose job is really to help people doing clinical trials meet all the requirements without necessarily having to themselves have to do all the footwork. So those are just some of the examples that we have put together at the Cameroon Campus Research Program.
Thank you, Sam, for sharing that experience. And it does sound very impressive. And I think that's why we see good results from the IDEAL program, because their scientists are supported very well and they concentrate on doing their science, as you said. So we'll move on to Gomo, I know you've set up the first support office at the University of Zimbabwe. Can you share with us how that was received by the academics and how it's supported?
Thanks, Alan, and thanks for this opportunity to share these experiences. I think we are all aware that the 5 key resources of a research system are essentially the human, the governance, the physical, intellectual, and financial capacity. And in our university, the University of Zimbabwe, and I would like to believe in most other African universities, integration of these is primarily focused on delivering on teaching at the expense of building the intellectual capital. So what we did in recognition of this was to try and change the paradigm where we realign the combination of these resources to be able to nurture an environment which encourages research. So we established what we call the Research Support Center. The idea is basically trying to address what Sam has said. How can we free time for the researcher so they can concentrate on thinking? Because the researcher needs time to be thinking, doing the science. So we established the Research Support Center to be able to provide the typical pre-award support and post-award support. What we mean by that is we firstly wanted to make sure that the researcher can easily be directed towards where there is possible funding in view of the very limited local funding, especially government funding. There is very limited investment in research, I think, in most African countries. So it's about identifying funding opportunities, disseminating them in a targeted way, and then, of course, you need to support the researcher to do the grant writing. And one major challenge is that the grant writing skills are fairly limited. So there's also a component of training that you need to do in grant writing. And not only that, because the systems and the culture have not been very proactive in supporting researchers. We have very few members of faculty who are engaged in research because partly they don't have the knowledge and skills to do that research. So there's quite an elaborate program that we have put together to do training in research, proposal writing, research methodology, the typical research courses. And then, of course, one big challenge is that the systems in our universities, in terms of financial management, are so bureaucratic that it's difficult to meet the compliance requirements of funders. So we also have yet to develop our financial management systems geared at efficiency and meeting the requirements of funders. So in a nutshell, we have tried to build pre- and post-award support for our researchers. And particularly for junior faculty and students, we have also had to put in what we call stimulating initiatives to get them interested in research. And this has included small research grants, fairly competitive at undergraduate level and postgraduate level. So these are some of the initiatives in a nutshell that we have been doing at the Research Support Center.
Thank you for that. Now that we've heard from all our panelists, I request the audience to start preparing their questions, which we will answer in the last 10 minutes of our discussion. But to continue this discussion, Professor Gomo, you did mention something interesting about particularly junior faculty. And I think that speaks to trying to feed the pipeline of research at universities. And so could you tell us how, apart from what you've described, what much more has the university invested in feeding the pipeline?
Right. So what I've described is the general support we provide to both members of faculty and to students. But to junior researchers specifically, we have initiated a number of strategies. One of them is the small grants that I talked about, but also through research capacity strengthening grants is also to introduce research fellowships at master's and PhD level and postdoctoral level. what we have found very interesting and what has encouraged the young scientists to come forward has been the training in research. And what we have done is to institutionalize that training. For example, all our MMeds are required to go through research training if they have to graduate. So we are evolving an institutionalized system for research training, and that has really stepped up our efforts to support students. The other efforts that we have put in place have been to engage scientists in the various disciplines in multidisciplinary initiatives where they are exposed to different disciplines and their efforts. And also thanks to initiatives like DELTAS and its predecessor, the African Institutions Initiative, and many other initiatives we're involved in. The bringing together of students, as has happened here, or fellows, has made a huge difference in the appreciation and the desire to conduct research within the young scientists. So these platforms are absolutely critical in engendering a culture of research among our students.
Thank you for sharing that. Graham, I'll come back to you. You did mention quite some huge figures of $600 million for regional projects. Could you tell us, how do our scientists convince their institutions to, plow into that so that they also do create their centers of excellence? How do they go about that?
So I think it's been said on the panel is that research doesn't just happen. You have to have the institutional structures that exist to support the research enterprise. And we often think about research funders, and then we think about research management as being about managing the funding side of things as well. But it's much more than that. It really is about the capacity to conduct research within an institution. And that involves training how to do research, how to write proposals, how to put in institutional review boards. Everything that goes along with the research enterprise is part of the project as well. Ultimately, we want to free the academic researchers, faculty, and staff to spend as much time as possible on the research activities. But that means we have to have the professional staff available and the professional resources available to support the overall research enterprise. So I think looking at the professionalization of research management, ensuring that it's not just an individual at an institution who is in charge of that, but rather the institutional structures in terms of policies, practices, offices, resources to support those offices go beyond an individual who may be championing them for a short period of time. This is what successful research institutions globally have in place. It's not just the researcher, it's the environment in which you're conducting the research itself. It goes beyond the individuals who are associated with it.
Okay, that actually now brings us to a very interesting aspect about the research environment that you've mentioned, the policies, the staff, the structures. So just say if you could, in your experience, how is it best to, set up the support structures that have been mentioned by Graham at universities, and how do researchers get involved in convincing their universities to set up these structures to support them?
Thank you. So that is, it's a two-sword answer, I guess, is the way. Many researchers don't actually want any support. They actually like to do things on their own. And so those are the ones that we have to try to convince that, it's very important. You can free your time so much more if you had that type of support. I've run into those types of researchers before. But then there are the others, of course, who welcome the support, and it is those institutions that are the most successful, where we have a majority of those types of researchers. So setting up the structures, it's usually an institutional decision, an institutional commitment to strengthening the research environment, and this is usually at the Vice Chancellor level. And in general, you would have either a DVC for research that's taking leadership of this, or the DVC for academic affairs. Whoever is at the leadership level responsible for research at the institution would be the champion overall at the institution. In a number of our institutions as well, we have different ways of structuring. And two ways, for example, that I've seen very common is where there are dedicated staff for the research structures or for the research function. And in many cases, because of lack of resources or for whatever reason, we also have academic staff members doubling up as the research staff in the research office. Personally, I think that having dedicated staff is the most important thing. Having your researchers doubling up doesn't effectively create that environment that you would expect and that you would hope that would provide the type of support needed, because they themselves are trying to do their own research, they're trying to teach, supervise students, and so it doesn't really work. So having a dedicated office for this function is one of the critical first steps. And I think we can talk more about this, but so first step is putting the structure in place, dedicated staff, having the different functions highlighted. What do you want to do in that research office? So we talk about pre-award, we talk about post-award, those types of things, identifying funding opportunities for staff, helping staff to write the proposal. Not the technical part of the proposal, but there are some key components that that the institution can help to support writing, helping people to identify key partners that are needed, budgets, always a problem, also contracts and grants, looking at the contracts, we need a legal team to look at this, IRB, looking at research integrity. So there's a whole range of things that is needed in that research function. Now, a small institution might look at a smaller number of staff, and larger institutions, obviously, would need larger numbers of staff to be able to support the function. But for now, I can stop there and we look at other answers.
No, thank you for that. I think that describes what I think institutions should have. But interestingly, to say that some researchers are hesitant to have support, we'll see when the audience starts the participation. But now to Sam. So in trying to create this environment that is conducive to your researchers that you're building, how do you balance the issue of equity versus excellence, and how do you then select finally the researchers you intend to work with?
I think this is obviously the old age conundrum of equity and excellence. And I don't think there is a very simple answer, because whichever way you lean, you have a risk. So if you want to be purely elitist, in an ideal world where everybody had the same level of education, the same level of exposure, then clearly you would just simply use academic merit, and that would become your simple measure. On the other hand, if you simply went for equity in a setting where, people haven't got the same level of exposure or academic background, then there's a risk of simply trying to tick off boxes and ending up with mediocrity. And so it becomes a real challenge. And I think there are two levels at which you can think of addressing that. The first one is in the short term. And let me say, for example, in Kenya, we have areas that we call marginalized areas, and this is probably true of all African countries. And then you have marginalized classes of people, and then you have marginalized gender. And in the immediate term, I think you try to address those by ensuring that, for example, in your recruitment, you deliberately go out to to recruit people or to target people from those areas. And maybe you, in a sense, you really work hard to try and find the best, but specifically from those areas. So in a sense, you try and maintain excellence because you're picking the best people as much as possible from the marginalized groups, but at the same time, you are trying to really go for the best from those groups, because regardless of how marginalized they are always really good people. The risk, of course, is if you simply want to do it for the sake of taking off, you may pick people who cannot be competitive. Science is competitive. You know, I may pick them in Cambridge, but I can assure you when they go for interviews, when they go for grant competition, people will be ruthless and brutal. So you've got to balance that. Now, the more longer term one, I think, is how we, for example, for us, obviously we're interested in science, we bring science to our citizens at a very early level. And I quickly can give an example of the activities we are doing at Ken Wellcome Trust Research Program to try and do this. So we are located in Kilifi, which is one of the marginalized zones or regions of Kenya, and we have what you call a school engagement program, where we are working with secondary schools around the county, and we are taking science to them. We have different activities that really try to expose them to science. And not all of them will become scientists, not all of them will necessarily improve in their science capacity, but the few who might never have thought of science as a career option, we see them turning. And we then have, when they finish school, the top student, O-level students in Kilifi district, we take 10 of them who come and spend 2 months with us. We have opened our doors to students from different universities. And the difference between us and a lot of other institutions in Kenya is that we give stipends to all our attachments, even when they come for industrial attachments. And the reason we do that is that we want to pick people on ability, not ability to pay. And we have seen that that makes a big difference. We'll get students from all corners of Kenya who would never have been able to come and live in Kilifi for 3 months had we not been giving a stipend. So that is some of the ways we feel that we are able to address. We ensure that people are not disadvantaged because of their economic or even exposure by starting to address some of these areas quite early, as much as possible. Thank you.
Thank you, Sam. You bring in a very interesting aspect of inclusion and trying as much as possible to bring in the marginalized communities. And I think that's a very commendable thing that you're doing at Kemri Welcome. You know, you did mention the ability, not the ability to pay. That's quite interesting to look at things that way. But I want to throw a question to Tom, who's the Director of Programs at the African Academy of Sciences, to really follow on the issue of equity versus excellence, and some of the things we are doing at the Academy to address that. Because in my interactions with many of the people in the audience, the question has always been, how do you determine who to take on? Yeah, so Tom.
Well, thank you for catching me unprepared, but that's... So I guess one of the discussions we've been having with the DELTAS directors actually this week is essentially what Sam has articulated very beautifully. The question of how do you make sure that the limited resources and the opportunities that are limited that the Academy is putting out are not just going to those centers that actually are centers of excellence and are always the top tier institutions on the continent. So I think the DELTAS model provides us with that opportunity of balancing excellence, merit, and equity, in the sense that we're talking about lead institutions taking the responsibility to bring in partners and to capacitate those partners. But more importantly, I think it's that question that Sam again has alluded to of, you can recruit the best people from all over the place. And so in Africa, we can group in, places around, we can say there are strength in the east and the north and the south, but perhaps not as much strength in the center of the continent. So in terms of marginalization, how do you then begin to ask the DELTAS leaders and institutions to think a little bit more about how they can recruit from those countries we are not seeing too many DELTAS fellows coming from? I could mention many of those countries, but I think you are familiar with those countries at the center of the continent where we think we can recruit more, and again pursuing that excellence model, not just doing it from a position of equity alone, but the balance of, of merit and equity.
Thank you, Tom. I wanted to see if you're listening keenly, and you are indeed listening keenly. So I would like to invite some questions from the audience, and while we sort that out, I would like Graham to briefly comment on, what transformations have happened in the centers of excellence that have been established wherever they have been on the continent. Just briefly, whilst the microphone moves to whoever has a question.
So I think one of the nice things about the Centers of Excellence program actually alludes to something that Tom just said, is that these are regional projects. So there's an acknowledgement that not every country has excellence in research in a particular area. So let's use water as an example. We have, out of our 40 centers in West Africa, we have 3 centers that are focused on water research. But they are required to recruit students from across the region. And when I say the region, I mean anywhere outside their home country. If they can recruit students from Europe, fantastic. Some from the Middle East are already coming. So I think the idea is that you bring students to the Center of Excellence, and then following their training, earning their PhD, that they're getting a PhD, then they return to their home country. And what that does is it builds up the capacity across the entire region in terms of that particular area. And it also builds a network as well. So you have water researchers who maybe are returning from Burkina Faso to Benin or Cote d'Ivoire or Nigeria. They have the academic network, they have the professional relationships, and that extends not just in the academic world, but also in the private sector world as well. You're building up a network throughout the region as well. We have an issue in terms of sustainability. These projects are 5 years, and I think this is an issue that comes in when we talk about access and sustainability. Academic or institutional sustainability in the university world means revenue generation, and that comes either from a national government, or it can come from private sector, which is limited, or it can come from tuition fees. So we have a balance between looking at tuition fees to generate resources versus stipends to make sure that anyone who is qualified and able can participate in the obtaining an advanced degree. It's something that we continue to struggle with.
Thank you. So there's a question there. Yes, please go ahead.
So Nadia Kelef from Institut Pasteur. Thank you very much to all of you first, especially on the perspective of not only building capacity from the human resources point of view, but also the infrastructure and the research management, which indeed are very important. Thanks also to Tom, who actually anticipated my question and built what I'm going to keep building upon. I could not agree more to what he said. And I would actually like to extend the question or the comment to the fact that I truly believe that according to the already existing community of excellence research center, which indeed include the one that you're pointing at, the World Bank, the DELTAS, and the Institut Pasteur network, I think there's not enough of recognition of the already existing excellence in Africa, as well as in low-middle income country. I think we're only seeing and funding the tip of the iceberg, and there's much more than that. So the challenge, I think, for the already recognized centers of excellence is how to bring more of the less visible or less recognized centers of excellence. Of course, I'm preaching for my own network, but also for the other institutions that are around the country and are good, maybe not as good in leveraging the resources around, but how do we work on that next step? And I'm going to try to be very politically correct, but I think we reach a level of indeed development of the research institution landscape where we should keep going in the direction of keep building instead of starting asking some of these organizations to deliver research on investment. I don't think we'll be saying that for higher income countries. And there's a momentum that we should not break, asking for the next step to arrive too early when indeed, again, regional is very important. The continent is big. The resources are already that have been invested are strong, are big, but there's much more that's needed to really reach the next level, and we should keep going in this direction, to my point of view.
Thank you. Whilst the panel thinks about how do we work to achieve the next step, there's a question there, please. Oh, yes.
Yes, thank you. The panel very well laid out the importance of having a research environment that is conducive for researchers to do their work. Unfortunately, most of the research programs, especially at universities, are private-public private partnerships. Usually you have a high-income country university working with a low-medium-income country university, and then they set up their own private research group. The university per se may not have a lot of input in this. When it comes to overheads, some research funders ring-fence the overheads and say they can't do ABC. I think a time has come when the people who support research, the funders, and the institutions and universities where research is done, come together and come up to be able to set up the system you're talking about. And my question, would DELTAS put this on their agenda to be able to kickstart this so that we set up research management units within universities and institutions, as opposed to each group having their own administrative system? Okay, I with another university, you with another university, and so you set up quite a number of systems. Space is limited at universities, so it makes it worse when each research group is trying to look for an office to put their administrators in. So I think someone has to break this impasse so that we set up the system that you're talking about. Could DELTAS do that?
Whilst we think about whether DELTAS will be able to institutionalize support systems at universities, we'll take the last question and then have the panelists respond before we move on. Yeah.
Thank you. My name is Shweta Khandelwal and I come from India Alliance from Delhi. My question is about the inter and intra institutional competitiveness, if you like, which sort of becomes very negative sometimes, in the sense that we do not get opportunities to collaborate internally or with other institutions when we compete for big grants or things like those. So who should take, in your opinion, lead on addressing these things within an institution? Like, should it be grants management? Should it be a different department? Who should see, like, who should buffer this out and tell you that you should partner with these rather than compete with these and things like those?
Thank you for those questions. We'll take the next set of questions after the panelists have responded. So maybe just to recap, the first question was about how do we work on the next level, not to break the momentum that we've built currently. The second is about institutionalized support systems. And the third is about intra-institutional competitiveness. Who should take lead? Who should be watching the different groups within institutions? I'll start with you, Graham. 2 minutes, and then Jose and Sam and Gomo last.
So let me address the last question first in terms of intra-institution. And this will come from a World Bank perspective, is ultimately the World Bank is making a loan to the national government. So it's actually up to the government and in some sense to the institution to decide what their priorities are. Now that differs from a bank perspective to maybe another research funding organization. The next level, I think this is a critical element. We are, the numbers that we've seen today and this week in terms of the number of fellows that are supported by the DELTAS program, 1,400, I think was the number that was put out there. The ACE project supports a similar number of PhDs and several thousand master's students. Many of those are returning to an academic institution where they have a position already. And there's a strong need going forward for them to not just be teaching, because there's a huge issue on access for undergraduates in terms of teaching, but to develop the ability to continue the research that they've been starting as part of their PhD at centers of excellence. So building the networks between the centers of excellence, which may be a hub, and the spokes that emerge, and building up the capacity in those spokes is a critical element. That requires the funding to do that. And that is a big challenge, I think, that we have to be thinking about from a funding perspective going forward. We are dramatically increasing the number of faculty members who want to build a research program going forward. There has to be an opportunity by which that can be accomplished as well. And that is, I think, a big issue that we need to consider. Many of our centers we know are at flagship universities. Many of the graduates of these programs are going to be at flagships, but also at further national research institutions and also universities, and building up the capacity of those as well. How to continue the research funding, to me, is an essential element.
Thank you, Graham. So Jacente, 2 minutes to respond to some of this. Yeah.
Okay, I'll answer question 2 and 3. Question 3, looking at who is responsible for the intra-institutional partnerships. So if you remember, I talked about the research management community in Southern Africa, at least, and we believe this is global. ARMA also has developed something similar to what we've developed at SARIMA, looking at the competencies of research managers. And one of the competency clusters that was identified is partnerships and collaborations. So research managers should have that competency of being able to identify the relevant partners, whether it's internal, external, international, wherever, to be able to make their institution competitive. So in my view, I would say that the research management office, or the function, whatever it's called, should be responsible for doing that particular activity. And many research management offices does this successfully. Of course, there could be other groups. Some people have international offices that are separated from the research office, and they take that responsibility. But there should be that function at the institution to manage that whole process of partnerships. The second one is around having systems that are institutional rather than the group programs around support. Fully agreed that rather than having several groups having, research support systems, an institutional system that can provide this type of support would be the most recommended. I know that sometimes it's because funding is attached to a specific program, project, and so that group would have that specific opportunity to have that support. But what institutions need to begin to think about is, how can I bring those different supports from different projects into the institution so that there is a central, or in some cases, if there needs to be decentralized, if you are large enough, then those systems would work. In terms of an overhead issue, I think we spoke about that yesterday, Graham, about having overheads that actually reflect what is the true cost of doing research in Africa. That is a key problem. Several years ago, the Essence group of funders worked on looking at the true cost of research in Africa and helping institutions to cost their research. There's a whole set of documents around good practice on the true cost of research. And I think they're doing a revision of this, a version 2 of this whole process. One of the key things that we did when I was at the University of Botswana was to develop an overheads policy. And so this worked out for us what was the cost of doing research, what our overhead should be, and what our overhead could be for different types of funders, whether it was a private sector company or whether it was a government funder, foundation, or whatever, different levels of overheads can be then charged for a specific project. But very key is to have a policy, and very important to try to lobby to funders. And so those funders who are in the room need to understand that there are real costs of doing research in Africa. Most of the research that is global is done here, and a lot of the overheads are going to the international partners. I know, Graham, we talked about some of the history of that, but we need to be able to bring this really to a lobbying sort of kind of forum where the real costs are actually appropriated in the right place, I would say.
Yes, Sam?
So I completely agree with what Jesse said, so I don't think I need to repeat that. But maybe just to bring, with regard to intra or interdepartmental competition, then to bring just a third aspect. So if you think about organizational structure, you have the hardware, and then you have what you call the tangible softwares, which are the processes, procedures, and all that. And I think those two have been spoken to. The third aspect, which I think is really key and often not really emphasized on, is what you call the intangible software. And that is the research culture, collegiality. So although you can have structures, you can have an office that says who should collaborate with who or who should compete with who, until there's that sense of a common purpose within, say, a department, and that people feel comfortable with each other to compete, to collaborate, then often what you end up with is competition. Of course, competition is good to an extent until it becomes toxic, and then rather than bringing up the institution, it ends up pulling down the institution, and you have all this siloed way of working within the departments. And I think a lot of that has to do with the leadership of the institution. So it's not just the management office, it's the way the institution brings together. So I think that's something that often is not really focused on, and that's what often makes a difference between departments or institutions or schools that are really progressive and productive versus schools that are simply trudging along. With regards to the next level, there was a question, I think it was from Harriet Mayanja, regarding whether DELTAS can fund the next level or can fund the development of these offices. And I think it's important, even as we are sitting in this sort of meeting where we are all very grateful to DFID or UK Aid, and we're all very grateful to the Wellcome Trust, to remember that the burden is so huge and has to be taken up by our own governments, the African governments. I think it's too much to ask the Wellcome Trust, too much to ask DFID, to be the ones to reform African universities. They can provide some catalysts, they can provide some seed, they can provide nucleus, but I think at the end of the day, this has to come through our African institutions. And please don't tell me there's not enough money. When I look at the Range Rover that my VC drives, that is enough to run a research office comfortably with everything that you need. The fuel alone, when they turn on the key once, that's enough money for the messenger. Turn the key twice, that's enough money for the... So there is enough money. I think we appreciate the Wellcome Trust, we appreciate the UK Aid, but we really must also ask our government to prioritize. You go to, sorry, I'm maybe taking long, but you go to any boardroom in the universities, they have the best chairs. You go to the next room, which is the lab, it doesn't have a microscope, it doesn't make sense. Honestly, go to any boardroom, university boardroom, and tell me what kind of chairs you see there, and then go to the lab next.
Gomo.
Thank you. I can only concur with the other panelists in terms of emphasis, especially what Sam has just said. But just to respond to the second question on the institutionalization and perhaps partly the third question on leadership, is that I think for us to be able to accommodate the next level that has been mentioned, research support or research management must become a systemic part of the research process. I think that's critical for if we are to move forward. The reason is because my experience is once you start developing or strengthening research management in an institutionalized focus, you stay a hornet's nest. essentially you have to deal with issues just across the board, which makes it very difficult for our local institutions where we do not have training as research managers, which I think Jose touched on. So you want to look at issues of governance. For example, our university did not have a research policy in the first place. So you have to look at issues of skills development. You have to look at issues of partnerships and stakeholder management. You have to look at issues of supervision and management. So I think unless we make all these processes speak to each other in a coordinated way, we will be talking about this for a very long time. And the other thing is, therefore, we need somebody who facilitates, perhaps not necessarily leadership. We probably need facilitation. These various initiatives can have several champions, but we need somebody who coordinates that. And I think that's the Research Management Office that can coordinate these efforts. So I think in a nutshell, we need our institutions, and to follow on what Sema said, we need to, in fact, one of the lessons I've learned is that where there's a will, there's a way. And we can only climb the hill if we desperately want what is behind it. So I think we have to put our act together as African institutions and take this forward, but also then get the support from those who are willing to give a hand. But we have to take ownership of this process. And thanks to DELTAS, at least, the REMPRO program, I hope, will develop some generic standards which will enable us to say, where are we and where do we want to go? Thank you.
Thank you, our panelists. Unfortunately, we've run out of time, so we're not going to take any more questions. But I think you've realized that what I said in the beginning is actually true. Research management systems are fun, and systems strengthen science. So as you've heard from the panelists, I'll give each panelist just 1 minute to quickly round out their thoughts and a key message for the audience to take home. Starting with you, Gomo. 1 minute, please, and I'll switch off the microphone.
Okay, thank you. It's a difficult one, but I think we need to think big, but act strategically. We need champions at the highest levels of our university leadership. We do not want to reinvent the wheel. We have a lot of things that are going right. Let's exploit what is going right. We do not want to fix anything unless it's broken, so let's try to utilize what we have. And finally, money is not everything. It's just a means to an end. We need to develop the ethos. We need to build the ethos within ourselves. And this is something I learned from some meetings in East Africa. If you want to run fast, run alone. If you want to run far, run with others. Thank you.
Thank you very much. Sam?
I'll just say research management matters.
Wow. Okay, Jose?
I don't know if I need to say more after Sam. But I guess just to say that there's a change in landscape in Africa. African institutions are now taking the lead in doing research and managing their own research. And therefore, the institutions must take on the mantle of managing this efficiently and successfully, and this can be done. There are lessons to be learned from many different institutions. There are already many different templates and things that are out there that can be adopted for the African setting. We just need to make sure that the African context is taken care of, but institutions can be successful. And we need to make sure that perhaps funders can put the pressure on institutions. Maybe we should be saying to funders that if an institution does not have the support system in place, then you don't get access to funding. And maybe we will see some changes at institutional level. Thanks.
Thank you, Jose. Graham, 1 minute.
Well, thank you. I'm glad I had a couple minutes to think about this. I think one of the things that Jose said was very important is that overhead or indirect costs are a percentage of the grant costs. So actually, as you build your research enterprise, the research management costs and the needs within a research management unit will actually increase. So I think that's something to be aware of as research capacity at institutions grow. I think the other thing that we need to focus on is that research is a key element of a university. Teaching is definitely there, but also the research enterprise, especially for a successful university to make an impact, is a key element to do that. So we have to have the support structures in place to do that, and the ability to translate that knowledge, not just into financial gain, but also into policy and economic development, social development for a country is a key element of the research enterprise as well.
Thank you, all our panelists. The messages I've received is the landscape has changed. African institutions get direct funding and therefore should be able to manage their finances and their research efficiently. Where there's a will, there's a way. We think big and act strategically, and research management matters for the research enterprise to flourish. Thank you, all our panelists, and I think they deserve a big hand clap. Okay, thank you, and have a good afternoon.
Yes, while they get off the stage, we can appreciate them for an excellent job. How many factorials? 5. Let's go. Good. Let me invite the following people to come to the stage. We'll have some rapid presentations before we break for lunch. We had thought we could have some of them now, break for lunch, and then reconvene. But I think it would be a lot better if we push and then we break for lunch at about 1:30, so we don't have to reconvene here, and then we go straight to the breakout sessions. So I invite Tom Kariuki, Grace Moara, Michael Kilpatrick, Shweta, I hope I pronounced the name well, and Olivier Naon. This name has always been a very difficult one to pronounce. I don't know if it's Vutas or Wutas, but Olivier, you can join them on the stage. Yes. So while the others are coming, I think Tom is loaded and ready to go.
Right. So, I don't know how many of you remember the true meaning of DELTAS. Can you pronounce it in full? Some people just come into the community that it is DELTAS, you take that up. But I want to talk a little bit about the L in DELTAS, which is for leadership. And then I will invite my colleague, Grace Maura, to develop this a little bit further in the context of what we are doing as an organization. So the L in DELTAS is actually for leadership. And one of the things that I would like to pose, especially to the DELTAS fellows, is this set of questions for you to think about in terms of your leadership development. So, and these questions are also setting up the expectations that we have as we try to shape DELTAS too. So we're thinking more and more about, have we collected all the key indicators? Do we need to revise our M&E framework? Do we need to revise our theory of change as we think about the future? So first question is the bar for leadership and standards of excellence in your own university, as we have heard from the last panel, or at your own department, at what level is that being set at your institution? Is the bar too low, or is it high, or is it at the right place? So that's something for you to think about in the context of everything you are hearing this week. A second question is that when we look at the more than 1,400 DELTAS Africa Fellows, how many of you will make a successful transition from being early to mid-career to really develop into research leadership, given that you have excellent examples as the leaders of the DELTAS networks, people you can try and emulate? How many of you will make that transition moving forward? And what do you really need to make that transition?
A third question is around what are you doing about your own leadership development? Because I think you can be proactive about going out there and actually seeking out the training and the mentorship you need, as you hear from Grace, to really begin to think about your own leadership development. And the fourth question is, as you make these transitions from early career to mid-career to senior leadership, are you doing more of the same? Or are you doing other things that are really adding value to your own career and to the societies and the big aspirations? Are you still stuck in this rut of grants in, papers out, seeking promotion? Or are you taking true steps towards your own leadership development? So I think the ask for when you are an early to a mid-career researcher is quite obvious. The expectation are that you work as hard as you can, and within that, you get supervision and you get mentorship. Everything you do is centered around the number of grants, the number of papers, and as you move towards mid-career, then you begin to think more about the number of students you can supervise and how you can work with your leadership to help in the supervision load, as we've had in the course of the last 2 days. But as you make this transition, and this is where we're thinking more at the academy about whether we are capturing the right indicators, then I think we begin to ask more of you. There is a balancing of teaching and research. Increasingly in our universities, it's a challenge for many of you, I know, given the demands of expanding student numbers, for you to make the time to do the research. So one of the things we have had to think about at the academy is for example, the work we are doing with the UK Royal Society around postdoctoral support is a question around protected time for researchers and the balance of that. How much should we recommend as the time you should take to focus on your research vis-a-vis the time that your university demands of you to undertake teaching? And when you are a DELTAS leader, it's not just that, it's also managing resources, managing people, managing facilities, etc., etc. So that's really key. Leadership transition demands that you take on increasingly other things around committee work or participate more in the peer review process. So you get invitations as you become more and more visible to participate in this kind of work. And there's an expectation that you should be maybe involved in editorial reviews or journal reviews, or as the AAS, we invite you to be a reviewer in a call that we have put out, as we have reached out to many of you to do that. Then there is a service to professional societies that is part of leadership development, because as Charles Rotimi said this morning, he was president of the African Society for Human Genetics for 10 years. That is a service to a professional society. So sometimes we are comfortable just being members of professional societies, but that is not sufficient. You can't just be a member or a fellow of the African Academy of Sciences as all you do is to put after your name FAAS, for example. There is more to being in that space, and you need to actually play your roles. Speaking engagements, how do you get invited to those speaking engagements, whether it is a meeting like this one? Can you organize conferences where the grand challenges, issues around your science can be discussed? How do you view the transition of your research towards impact? I think the focus on research questions is particularly important because leadership demands that you are fully aware of what is the state of research in your chosen field of research. What are the big questions that you are pursuing? And are you actually able to then organize meetings that actually where the articulation of those big questions are happening, and then moving forward with your research and your research group to address those questions? Because you have a strong CV and you build a strong CV, you get more visibility. And again, like has been emphasized on this panel, you can't get away from issues of scientific citizenship and the collegiality that is required of a leader. Think about writing commentaries. Commentaries are great, and putting out commentaries, especially around the big research questions, it's one way for you to raise your profile as an emerging leader Others will read about you, and they will know you beginning to get into a space where you can make commentaries. So for DELTAS Africa, we want to devise and revise our M&E frameworks to keep asking the question whether we are able to track all these outputs and outcomes moving forward. So that's something we are thinking a lot about phase 2, including what really we mean by mentorship and the services we can offer in that regard. I think the final thing I want to say, I came across this and I really liked this quote from Kim Cobbs, who is a climate researcher, and she was talking about the criticism sometimes we face as we make this transition. So I hear from colleagues that others say they disapprove of my interaction with the media and what they call my advocacy. But I have tenure and I'm an endowed professor, so you're saying the rest of my life will be about impact with a capital I. So I think that's a very confident person who have arrived at a space where they feel they can now provide the true leadership and want to emerge as a true leader. Not everybody may choose to go in that direction of saying they want to just have a focus on impact, but we all can emerge as leaders and get to a leadership position through various ways, but we all have to try and choose the path that we want to follow with regard to that. So just wanted to offer those introductions to this topic of leadership, and then invite my colleague Grace Maura to really talk a little bit more about our plans for mentorship and leadership development. Thank you very much.
Good afternoon, everyone. Thank you. I can't see something. Thank you. So I'll briefly take you about through the mentorship program, but before that, I want to highlight several other programs we have at the Academy, and overall we refer to this as the Rising Research Leaders, and this is one of the 3 objectives of the ISA platform within the AES. And so the specific goal that this falls under requires us to identify and support early and mid-career researchers to stay and build their careers in Africa. And it's not just staying and building your career, but it's actually thriving in that career. And at this, we are looking at postdoctoral researchers and postdoc. So this is beyond your PhD and beyond your postdoc. What else do you become? Do you actually thrive as a researcher? Do you become a leader in your field? And so we also expect you to be setting the research agenda on this continent. And so how best can we support you in that? How best can we support you to achieve all those things that Tom has been talking about? So we have a couple of programs at the AAS where we support that kind of work. And what you see here, you will be able to, the pointer is not working, but the table you see there are the various programs that are there. We have the affiliates program, which falls right under the first mandate of the Academy, which is recognizing and promoting excellence on the continent. And here we recognize early and mid-careers under the age of 40 who are already excelling in their science in all the disciplines, and they are based in African institutions on the continent. We've been selecting these for the since 2015. We now have 4 cohorts. We have a total of 87 spread across all the regions of the continent. In fact, on Monday, we closed the 5th call, and we had quite a good turn-up of about 368 applications. We had to look at them, but we're really hoping we can keep this momentum of actually letting the world know that actually we have excelling scientists. And so that's a major group of how we recognize the work that you're going to be doing beyond your DELTAS fellowships. We have APTI, which is the Africa Postdoctoral Training Institute, which is funded by the NIH. And for this one, we have postdocs going to the US for a period of 2 years and then coming back to Africa for another period of 2 years where they're supported to do research in the health space. And then we have SACO, which was working on climate change, and this one came to an end. We had master's, PhD, and post-PhD fellowships. Then we have climate research for development, which is ongoing. We just awarded last month, and they will be doing research on various aspects of climate change, and also there's spread across the continent as well. Then we have FLAIR, which is funded by the Royal Society and the GCRF. And again, these are postdoctoral fellowships enabling them to do research in the natural sciences for a period of 2 years, maybe could extend it. We're yet to see how that goes. And then we have the RISE fellowships, which are about 7. But all these terminologies mean something, so we don't come up with the words just like that. And these are, again, other 7 fellowships in the natural sciences. So what I'm saying here, by just highlighting these fellowships, is to tell you that beyond the DELTAS fellowships, once you complete your PhDs and your postdocs, there'll be various other opportunities that you will find out there to enable you to continue doing the work that you do and to enable you to support. So what do we do with these groups of researchers? So the affiliates program, which I specifically talked about, I want to highlight what actually these individuals benefit from, and you have a few of the affiliates in the room. I think we have 3 or 4 of them who are also DELTAS fellows. They mostly will benefit from individual career development. They'll benefit from mentorship, from training opportunities, networking, and collaborating activities, because we do realize it's important for you to do this research, to have a well-equipped laboratory, but we also know that it's very important for you to be within the right network so that you can actually find the right people to collaborate with on a research grant, the right people to enable you to communicate your work better, the right people to talk to so that you can actually engage with policy makers. So these are the various aspects that we focus on the affiliates program. Then we have the Future Leaders Africa Independent Research. This is the FLAIR program, as I earlier mentioned, and our fellows mainly focus on the 3 key grad challenges as per the GCRF, and just have a group photo of the team when we did the induction of the first cohort this April in Nairobi, and you can see the various fields that are covered. So if you are a DELTAS fellow, you may want to consider applying to future grants when the call opens out in the future. I think we already have one ongoing or has closed. I think it has already closed, yeah. And then we have the African Postdoctoral Initiative, as I earlier mentioned. This is currently ongoing. We have a team already selected, 10 of them already selected. And then we have the Regional Initiatives for Science Education, also currently ongoing. And again, you can see the partners that we are working with on various of these programs. The Climate Research for Development, and I think this is very important because the outcome of this project, what these researchers are going to produce, we are hoping is actually going to feed into the African voice when it comes to climate change issues on the continent. So not just producing the research, but actually influencing policy. There's already a conversation on what contribution this could make to the IPCC reports. And so we are really keen to see the output of each of these researchers. We have a group of 21 researchers spread across the continent. So the climate research for development is very interesting because if you've been looking at the various maps that have been presented in the last 2 days, you will notice we're always saying that we don't have many researchers coming from Central Africa. But actually with, and also with this one, you see that we don't have many coming from Central Africa. But what is interesting is that whereas this grant is Pan-African, so we can select from every, you can see we still have many coming from Western Africa, Eastern Africa, and again Southern Africa, but very few from the Northern, again Central, we do not have representation. But it's also interesting to note that the Francophone Africa is quite strong when it comes to climate research, which for us is a good thing to highlight. But what do we offer on these programs? What do we offer when you say we are talking about the rising research leaders? There's the membership, which is the affiliates program, and then there are the fellowships, the various ones that I have talked about. But we're also working on a leadership development program for these early and mid-career researchers. So other things that Tom has highlighted in his presentation, how best can you deliver this? What kind of training is required for someone who has completed their postdoc and now they're looking to transition? How do you train them? How do you prepare them for innovation? How do you prepare them to manage a team? How do you prepare them to engage with policymakers? How do you prepare them to actually publish their work for the lay audience. So these kind of trainings we've been putting together, and we are mostly doing this currently under the FLAIR program, but we are hoping that actually we're going to expand this across all our Rising Research Leaders programs. We have just mentioned a few of them that are currently ongoing and some of them still in discussion. We also offer networking and collaboration opportunities. So the conferences that we organize for these teams are not purely science, but we try to bring in the science and the networking together so that we actually enable them to have that platform. Some of the things that people don't talk about is how shy some of the researchers can be and scientists can be, but how do you actually facilitate that process to happen in another way? Is it in how you organize the conference, in how you design the sessions, so that even the introverts can actually have an experience of talking to someone? So it's all about how you do these kind of things, and of course the scientific meetings as you have here, the DELTAS Africa, but also publishing support. So I'm sure all of you, the DELTAS fellows, you know that you can publish on the AAS Open, and also all our other Rising Research Leaders do get to publish on the AAS Open. So this is one of the contributions that we hope can keep growing. And as well, and finally, is the AAS Mentorship Scheme, which at this point I want to spend a few more minutes talking about, because when I joined the Academy last year, one of the things that we really needed is to connect our fellows, and these are the fellows of the Academy and our affiliates. But the main reason why that was required is because we needed to build a mentorship scheme for these affiliates. And it's also because we did not feel like people they had enough role models around the continent. Perhaps in the health space, as I can see with DELTAS Africa, maybe most of you do not have a challenge with finding role models in your field, but in other disciplines, you will struggle to get someone, for example, in mathematics who is excelling and whom you can interact with. So what we're doing with this mentorship scheme is, our vision is to actually have a critical mass of talented and motivated scientists producing, translating, advocating for impactful science on the African continent. And what we want is all of you here seated in the room, some of you will be mentors and mentees. We want you to be able to mentor someone else so that that person can eventually mentor someone else at a lower level or even at their level. And then we are building a generation of motivated scientists who are there to support each other so that we actually can achieve this critical mass that we are talking about. Most of the things that Tom earlier mentioned will be enabled through this mentorship program. So we've designed a distance mentoring program. We've designed a distance mentorship program where we will not require the mentors or the mentees to meet face-to-face, because we know that will have a huge cost implication. But we're also very much aware of how the technology is very helpful on the African continent. For example, we have been here 300 plus participants, but we also have been live streaming this event. So actually, we have people around the continent following this conference without having to come to Senegal. We believe that if we use the same model for mentorship, it's also going to work, because then we can enable someone to have a mentor in Canada, someone to have a mentor in Egypt, whereas they are based in Nigeria or Kenya or Uganda or wherever else. And we hope they can make use of these technologies that exist that are low cost and they are time efficient, but they also get the support that they need. So currently, these are the groups that are going to be benefiting from the mentorship scheme for the first cohort. We have the FLAIR Fellows, the affiliates, Climate Research for Development, the SACO and the DELTAS. Actually, this is not the first phase, but it's actually all the phases, all the groups within the AAS. I'm not sure if you can see that very clearly, but is this how the model will look like? We've already enrolled mentors and mentees. We have about 70 mentors already registered, and we keep enrolling mentors as we move along. So if you'd be interested to become a mentor on the AAS program, do come and have a chat with me, and I'll guide you through that. We've already enrolled the mentees, and they have been going through the materials that we prepared for that. What you see on the side, there are some of the materials that we have prepared for the scheme. Our mentors come from various categories: industry, research managers, policy makers. We're looking for entrepreneurs. We're looking for communicators who can become mentors on this scheme. Because again, if you go back to what I talked about the cohort that we are working with, these are not people who are starting on in their career. These are people who are actually trying to make the next step in their career, the next major step in their career. So the kind of support that they require, the kind of mentorship that they require is actually very, very specific. In other times, we'd actually say they're actually seeking more sponsorship rather than actually mentorship. So we are very keen to ensure that we get a whole range of mentors on the program. So we'll be monitoring this every 6 months, and even in between with the mentorship coordinators assisting in this, because we want to make sure that we are learning as much as possible from the scheme, and we're able to improve on it as we move along. Our goal and our vision is to make sure if it's working for the programs that are within the AAS, we can expect this to your institutions, we can expect this to your universities, and you can say, I have this group of students who need mentorship, would you offer this to them? We want to keep this pool of mentors motivated and available to your students, to your postdocs, so that they actually know, I am on this science career track, and if I need help of any nature, I know I can approach my institution to get me a mentor from the AAS. We are glad we have over 400 fellows of the academy, and we know this is very helpful for us to run the mentorship scheme. So as I finish up, the focus areas, as I mentioned, because they're diverse, you can say categorize them in five areas, all about the science, but also about leadership, about your own branding, your own personal effectiveness, but also emotional intelligence, work-life balance. It's how you develop as a whole person, not just as a scientist. So we're really interested in mentors who are able to offer that. And you can refer to our website for the resources that we have. So if you go to the AAS website, you'll find mentorship scheme, and you'll find the resources I have highlighted there. And this is how the resources look like, and there are a few videos on the YouTube site of the AAS, so you should be able to watch those ones. But overall, what we expect with these rising research leaders is that we want to produce world-class research and innovation, but we also want you to have good financial management skills, leadership skills. We want you to be able to communicate with all manner of audiences, but most importantly, we do want you to have impact on science, on society, as an entrepreneur, as an innovator, but also as a scientist. Thank you very much. If you have any questions, we can catch up later. Thank you.
Michael.
Good afternoon, everybody. Yesterday we were privileged to an awesome talk from Martin Chalfie on GFP, green fluorescent proteins. I can promise you now a significantly less awesome talk on GFGP, good financial grant practice, but I did something managed to rub off from Martin's talk, because the title is now fluorescing in green, so there was some advantage. I've spoken to this community previously about the Good Financial Grant Practice Standard, and so this is just an update as to what's happened since the last time we met. The standard is now available in French, Spanish, Portuguese, and Kinyarwanda. The self-assessment portal, which many of your organizations have been working with, the production version of which went live in February of this year. The certification scheme is the kind of ISO process whereby certification bodies, which is a posh word for auditors, become licensed to undertake site certification audits. The scheme is developed by a group of about 40 plus technical advisors in a technical working group. We're just convening the Scheme Governance Committee, which is an autonomous body, autonomous of the AAS, because it needs to be shown to be very fair and transparent. And the anticipation is that we'll be licensing the first certification bodies in September, October of this year, so there will be licensed auditors available to undertake site certification audits from the end of this year. The next strand I just wanted to really bring to your attention, it's become obvious to us that to become trained in all things to do with GFGP, we need to have what we're calling licensed training providers, and this is going to be a cohort of training organizations who have demonstrated to the AAS and to the Scheme Governance Committee that they have the capabilities and capacities to correctly train organizations to become certified to GFGP. We're currently convening a technical working group so that we can get the best input that we can as to how that licensed training scheme should work. So if anybody wants to volunteer, then please reach out to us. The objective is we should have our licensed trainers available towards the end of this year. We know our community. We're across 21 countries and 54 institutions. And out of those 54, 38 have completed the assessments of GFGP. I'm actually kind of reporting back the homework of the last 12 months, and some of this you saw earlier in the week. Of the lead institutes, so these are the lead consortia, 9 out of 12 have completed their assessment. So that's 75%, 4 at platinum, 2 at gold, 1 at silver and 2 at bronze. Now, to remind you, to be a lead institute and to be sub-granting, you need to be working in compliance to the gold tier. So we have a couple who've not done it, 3, and we've got another 3 who have not done it at the required level. If we look at your sub-grantees, there's a cohort of 42 of them across Africa, and here we're talking about the African sub-grantees. Now, 60% of those have completed the GFGP compliance assessment in the portal to the tier which was chosen. But what I found interesting is if we look at the non-African sub-grantees, of which there are 23, only 3 of them bothered to do it. Suspicions of Global North not liking what the Global South is asking them to do. They're quite happy to do it the other way around. In terms of timelines, this is where really you need to pay attention. What we're asking is that the Delta lead institutions are requested to refresh their assessments to the minimum of the gold tier by September of this year. So you need to go back into the portal, you need to refresh your assessments, upload your supporting documents. We're also asking that your sub-grantees are encouraged, and that's a word that you can interpret whichever way you like, are encouraged to undertake / refresh their assessments by the end of this calendar year. Now, we're cognizant of the fact that for Deltas 2, some of you may wish to change your sub-grantee cohorts. So you have to interpret which of your current sub-grantees you're going to ask to refresh or retake the assessment, and or whether you're going to be bringing new sub-grantees in. Now, this last bullet is the critical one. To be eligible for a Delta 2 award, your organization and their sub-grantees are going to have to be working in full compliance to the requirements of the standard by the time those awards are made. I'll be frank, we had an internal discussion with the AAS as to whether the Delta leads and the sub-grantees needed to be certified to GFGP, but we kind of softened it a little bit. So we're now saying that you will have to be working in full compliance to the requirements of the standard to be eligible to receive the Delta 2's awards. Just like to thank our funders and founders, and thank you very much.
Okay, Shweta.
Hello, everyone. I'm glad that we had this little bit of exercise going down and coming up. So the rest of you guys, I know maybe calorie shock from morning you're listening to talks. My name is Shweta Khandelwal, and I am representing India Alliance here. I come from India, Delhi, where I am heading the Nutrition Research Group at Public Health Foundation of India. I'm going to present an innovative model of partnership to strengthen India's research capacity and enhance the ecosystem, which is called the India Alliance. India Alliance is a public charity that invests in transformative ideas and supportive research ecosystems to advance the discovery and innovation to improve health and well-being. It's sort of a similar model as we are discussing in Seeing Deltas Africa. So this is an India-based model for research and partnerships to provide funding for researchers. It's not a long, previously established institute. It was set up in 2008, not even approximately a decade ago, but has managed to touch a lot of lives in this last 11 years of existence. It is an innovative funding opportunity, like I will show you, the fellowships and grants which it offers to build research capacity in Indian region. But it does not preclude people from different nationalities to participate in that, and we'll see that also in just a minute. It helps to catalyze internationally competitive research. India Alliance Fellowship objectives are, number one, implementation of a globally recognized grants funding model, development of effective mechanisms to nurture outstanding biomedical scientists. And here when we say biomedical, it takes into account the full spectrum, right from basic sciences to public health. This I bolded especially because I saw this particular reversing the brain drain objective in DELTAS Africa also, and we have a similar one here, which enables transitions to India, reversing the brain drain. Providing a pan-India platform to perform competitive biomedical research, as I previously alluded to, and devising new mechanisms to promote clinical and public health research under this fellowship. Like this chart shows you, we have several types of fellowships which are offered at an early stage of your careers. If you are trying to look for a launch in your careers, there's an early career fellowship which moves on to a little bit established researchers, which come as intermediate fellowship, and people who are looking to lead research groups can apply for senior fellowships. There are also Margadarshi and other kinds of fellowships now, the Team India Science Grants and others, which you can have a look on their website. The fellowship programs, like I told you, caters to a whole spectrum of research fellowships. Who can apply for India Alliance fellowships? People who have a PhD in science disciplines, an MD, MD PhD, MS, DM, MPH, or even equivalent degrees. So it's fairly flexible, as you can see. In terms of salient features of India Alliance fellowships, the first is the emphasis on the lead investigator. A lot of space and money and environment to be able to grow into a good, high-quality scientist. It has no restrictions based on age and nationality. I think this is something very important to point out. Many times, even women, for example, into your research career paths. Due regard to non-research career breaks, like just I mentioned, no requirement for a salaried position or commitment towards a position. So when you apply, if you do not have a committed position already, they're still going to support you and try and give you a fair chance of competition in the fellowship. It is an online, secure, and user-friendly application process. The India Alliance system, the grants management system, it's an online portal. Encouragement to pursue interdisciplinary research. This has been increasingly seen in the last several years of the applications. They are trying to more and more get people who are collaborating with different disciplines, especially in the public health area, for example, where you start from health systems, go on to economics, nutrition, things like those. There are opportunities to develop international collaborations and also have international peer reviews. And you get continued feedback on your application. This also was a little different than, one-time funding which you receive and you only submit an end report. They actually convene annual meetings and give you feedback on your updates and progress. They're flexible and generous, okay. India Alliance Fellowship Awards, so far in the last decade or so, the total number of applications they've received are about 3,000 and the success rate has been 11%. So it's fairly competitive, as you can see, and a very high quality. So about 350 or so have been funded so far, like I was mentioning, early career, intermediate, senior, or Margadarshi. The diversity, as I was seeing from day one, we are very concerned about the diversity in the research settings. So this is not doing too bad. We have 35% of women and about 65% of men as fellows. We also have about 80% of them coming from basic research and about 20% from clinical and public health research. This is something which we can further improve. In terms of 24% of the awardees working at new institutions. So it's not something which Tom was alluding to earlier, only the top institution or the center of excellence is getting all the award. They've made sure that we are going out to actually smaller and newer institutes also to be able to give awards. You may not be able to see all what's written there, but just understand that there are a variety of disciplines within this umbrella which they fund. And some of the examples of our work, 5 of us are here from India Alliance actually, Suparna, Gagan, Shankar, Khushi and myself. And we are happy to talk to you. These are our areas we work on. 2 of us work on nutrition. One is working on cardiovascular lipid profile and fat distribution. And 2 of them are basic scientists, one working in TB and the other on viral diseases. Anyway, so you can understand I'm a public health scientist, so I will just put it at that. In terms of applying for India Alliance Fellowship, I think the best will be to visit their website. It's very user-friendly, very self-explanatory. You can check your eligibility, and even more easier way will be to come and talk to any of us. We are right there. So thank you very much for your time and giving me the opportunity to come here and talk about India Alliance. Thank you.
Olivier. Hope you're noting down all the questions and the people who will answer the questions. If you haven't done so yet, please start doing now.
Great. Well, thanks for having me. I'm going to keep my remarks really short. I know I'm all that stands between us and lunch, and I have a whole session in the afternoon. So my name's Olivier Wouters. I'm an assistant professor at the London School of Economics. I won't actually be using the slides now. It's for this session. So this is really just a gentle request for as many of you as possible to come to the session. It's a project I'll be presenting on that is funded by the Wellcome Trust. It's being conducted in collaboration with the African Academy of Sciences. We've been trying to measure health science research capacity across 54 African countries. So it's been a massive data collection exercise. So in the session, I'll be running through some of the preliminary findings and actually asking participants to interact and engage with the data. So it's hopefully going to be an interactive session. So that's the 2 things I want to point out. If you want to come to the session, please do bring a computer or a mobile device so you can access the website. Again, it should be interactive and fun. And it's been actually moved to room CO5. It was previously scheduled for CO5. It was previously scheduled to be in BO6. So just note the room change. As an extra incentive, it should be pretty short. So I think we're going to be running around 1 hour. So it should be a pretty quick and fun session. But that's really it for now. Yeah, and I'll be able to go through, leave you in a bit of suspense so you can see our findings in the afternoon session. So let's break for lunch.
Wish all the presenters were like Olivier, but they have all done well. So we have time for questions, about 10 minutes, and then we break for lunch. Branwyn? And Professor Jimde? And Snowin? Okay, let's take the first 3 questions. So where's Jimde? Okay, and then Val.
I'll start since I've got the microphone. I've got 2 questions, one for Grace, namely how do you match up your mentors and mentees? And one for Olivier, I know you have the short, I'd still like to know a little bit of what the highlights are, because I'm not going to make it to your session later, sorry.
Jim Day?
Thank you very much. Jim Day from Belgium. My question is on the GFGP. I understand that the next DELTAS, if there is one, it will be, you require that all the lead and participating institution would be GFGP compliant. I think this is a wonderful intention, but it would come with the caveat of the consortia being obliged to drop some potential members. If I understand correctly, there is also an intention to open up the different consortia to have stronger institutions bring in weaker institutions so that they can reinforce them. At this stage, there would be institutions that would despite all the efforts, would be very difficult to bring to a level where they are compliant. And the only choice would be dropping them and moving on with ones that can fulfill these GFGPs. So if this is a requirement, it will decrease our ability to reach out to colleagues in Central Africa, for example, where bringing people up will be an uphill battle, which we are facing currently. Even for recruiting students in those areas, it is very difficult to, when you launch call, you simply don't get applications from these areas. That's why the representation is that low in these places. So for financial institutions, it's going to be the same thing.
Okay, Val, just behind you.
Thank you very much. Very, very interesting panel, thanks. Just I wanted to bring back the issue that actually came up at the end of the last panel as well and link it up. With the example of good financial grants practice again, I noted that it's now in Kenya, Rwanda, which I'm assuming is because the Rwandan government has picked up GFGP nationally. So I just wanted to ask our African colleagues here maybe what they can learn from or engage with our Indian colleagues to develop more in the areas you're all working in and talking about. What is the role of national governments and maybe also regional bodies like WAHO in this case, as we're in West Africa, in promoting the kind of areas of leadership and good governance that you've all talked about really eloquently. Thanks.
Let's start with Michael, and then we go to Grace, and then Olivier, and then Tom has indicated that he's got a comment on the GFGP questions as well.
Okay. In terms of requesting that all the lead institutes and their sub-grantees are working in compliance too, it's difficult. We appreciate that. But the counter comment is there's no point in having a standard if it's not used. Now, to make it sound a little bit softer, there is a requirement that the lead institutes are working in compliance to the gold tier because they're making sub-awards. For the sub-awardees, they can be working in compliance to the bronze or the silver tier. Now, as an example, we've got many, many bronze tier organizations which are small cooperatives. In Rwanda, Kenya, Rwanda, that's why we translated the standard into Kenya, Rwanda, so that these local cooperatives could understand what the standard meant, because it was kind of complicated English. They are now working in compliance to the requirements at the bronze tier of GFGP. So we sympathize, we empathize, but if we're going to build capacities across Africa, it's not just scientific capacities we need to build, it's financial capabilities and capacities. And there ain't any other way around that, I'm afraid. I do recognize that some of your sub-grantee consortia may be different for DELTAS 2 than you currently have. You may want to add more, you may want to drop some. And that's a debate you need to have internally. The Secretariat at the African Academy is there just really to support you to achieve all these objectives. We've been circulating around you the last 2 days. We are available to help, support, mentor, and take this community from where they are from a financial governance point of view up to where we need to go. If I could pick up Val's comment on the sort of governmental aspects. We're now getting traction for good financial grant practice across the African continent. It's been adopted in Rwanda. We're about to enter into an MOU with Uganda. Kenya wants to adopt, Botswana wants to adopt, etc., etc., etc. Because they are seeing the value of improving the financial governance for both governmental and non-governmental awards, which are made within their own sort of boundaries, as it were. What drove the translation of the document into Kinyarwanda is that for the local people in Rwanda where English is not their first language, they could not understand what the standard meant, because it's kind of complicated English at the end of the day. So it's an unofficial translation. It's not the official standard, but it gives them an understanding. And it reflects some studies we did in Kenya last year in the community-based organizations, where the people supporting the CBOs doing the assessments were translated into Swahili. So we think that for the community-based organizations in the rural environments, let's say, we might have to consider translating the standard into the predominant local language in those areas. Thank you.
Okay, Tom, you want to add to GFGP
question?
Briefly,
I think Jim Day asks a very important question. I just want to make sure that there are no contradictions. We will not use GFGP as an instrument for reward and sanction. That's not the point. So what we want to do is to collect the data. And so it might well be that you're collaborating with an institution somewhere in a disadvantaged geography, who then, once they do the assessment, they are at the bronze level. That does not mean that you recommend that you don't work with them. It means that we then have the data, and we can begin a process of working with them to lift them up. That is the plan. It is not to use GFGP as an instrument for dictating who should be and should not be part of DELTAS 2.
Grace?
Thanks for your question. On the matching process, for us to enroll a mentor and a mentee, the mentors, we ask them to state the kind of strengths and attributes they're going to bring to the mentoring program, and we ask the mentees to state the needs that they have, and we use that to match, to do a closest match. We check in with the mentor if they are comfortable with a particular mentee. We check in with the mentee if they are comfortable with a particular mentor, and only when they agree, then we put them, we match them. We are hoping if we get the finances, we are going to develop a platform where this can happen without the interference of the mentoring coordinator. But we do realize the critical role that mentoring coordinators play in mentorship. So even as we think of developing a platform, we do want to keep the role of the mentoring coordinators very key in the mentoring scheme. Yeah.
Olivier, you had a question.
Thanks for the question. Yeah, I'll give a bit of an overview. So the study or the project is comprised of two different parts. The presentation in the second is going to focus on the first phase, which was really just a mapping exercise to see what information could we collect on different indicators of health science research capacity. And this was broken down into 5 different domains. So we collected a lot of bibliometric data first for 54 different African countries on publication outputs, focusing on peer-reviewed publications. So for each country, the number of publications, or number of publications that are produced, that was counted as any publication where at least one author came from a particular country. We also looked at the number of publications where the first author came from a particular African country as a measure of ownership of research. We also collected data on different collaboration patterns, so the proportion of publications that are with international co-authors, just with national ones, or single-authored. So that was the first sort of domain, was around publication. The second one was around research and development expenditure, so gross expenditure on research and development as a sort of an indicator of prioritization of health science research. We also collected data on the number of researchers in place in each African country. So there's a second broad domain. So then we also try to collect as much information as we could on research capacity. So things like clinical trial infrastructures are in place, the number of trials that are being conducted, the number of patents that are being awarded. And then finally, information on research institutions and funding. So number of universities in each country. the number of WHO collaborating centers, the number of institutional review boards that are in place. So some of the topics that were mentioned in the last panel. So again, this was just really a scoping or a mapping exercise to see the universe of data we could gather to get some sense of the level of health science research capacity. And the last domain, sorry, I forgot to mention research funding. So the amount of funding that's being dispersed to researchers based in different African countries from the 10 major global donors. So this is the first part, so again, a mapping exercise. The second part was case studies in 9 different African countries to sort of to learn about experiences trying to grow and improve the health science research capacity from the perspective of different stakeholders. So that's not going to be the focus of the presentation this afternoon, but I can go into that then as well. Yeah. So there's no short answer also in terms of what the key findings are. It really was a comprehensive mapping exercise. Different countries are faring well in some areas and perhaps a bit less well in others. There's no sort of one single sentence answer. It's really just really a comprehensive overview of what can we tell currently.
Yes, Millicent, you have a question, and then I can take one more question, and then because I'm chair, I'll ask the last question.
Okay, thank you so much for the panel for such an inspiring presentation. And my question, I have 2 questions for Grace and perhaps Tom too, following the first presentation. So the first one, looking at the eligibility criteria for selection of the fellows into the mentorship program. And my question specifically is, how do you actually account for the fellows? And these are mainly the female fellows who've had to take sort of career breaks in terms of, first of all, managing the family aspect first before they came now back into science, and they happen to be beneficiaries of some of these DELTAS program. And as in, how do you account for that? Because I feel they're now sort of left behind when you put their age, like 40 to be the age limit. And the second question also comes into the mentorship. I see you really have a very nice mentorship model for the AAS. And in my work that I'm looking at in terms of looking at the equitable career pathways within the DELTAS with the gender lens, is trying to look at these, find answers to what are the likely attrition points for fellows across the science pathway in DELTAS. And what I'm really, in terms of some of the preliminary findings that are coming from it, we are likely to lose, especially female fellows at the PhD level at that point when they are now completing. Because the question that really comes, I want to finish this and I want to sort of start establishing my family. How will I manage this? And they're looking at it, they really don't really have right mentors, right models to really associate with. So if this mentorship program is really mainly focusing at the postdocs, I also feel the junior researchers will somehow be left behind in terms of, so how is there a possibility of this beautifully outlined mentorship program to be extended even to the MSc and the PhD levels?
Okay, thank you. Last question.
Thank you, Mr. Chairman. My question goes to Shweta, the Indian Alliance. Your presence here clearly indicates that you are willing to open this opportunity to African scientists, but all the aims are toward enabling research capacity in India. So my question is, what are the chances for any African to get any of this award? And do you have any African awardee? Or are there any unsaid rules such as collaborating with institutional partners in order to be funded? Thank you.
Oh, you just took my question. So Shweta and Grace, you have the honor to give the answers and we end the session.
Thanks, Millicent, for your questions. On the first one about the eligibility criteria, I have to say what I presented was quite brief. The call that we just closed, yes, we have an age limit of 40 years. However, we do have a clause that states that female applicants above 40 who can actually justify the breaks that they have taken will be considered for this application. And they have come back, they have come and asked those questions, and you have told them, go ahead and apply. But then I have to mention, as we've been mentioning these 3 days, that it is about how excellent they are. So we will allow them to apply beyond the age of 42, but they need to be excellent scientists because it's a really competitive program. And I believe you are referring to the affiliates program and not the AAS Fellows program. On the question about the attrition points for the female fellows and how to get mentorship to the PhD and master's level, that's a very valid point. And there's a reason why we have started with the postdoc and above. It's because of the various programs we have within the AAS. I am aware that various DELTAS programs actually have mentorship schemes, and so we know those are already continuing for the PhD. I know their Mari one, for example, has a mentorship program, and we do know some of the PhD and master students do receive mentorship on that. However, we are in discussion to see even our next cohort, we can first enroll the postdocs, and then we'll move on to the PhDs. However, how the model is going to look like, the current mentees will be the ones who become mentors for the PhD and MSc students, because remember, we are actually working on having a generation of mentors and role models on the continent.
Tom, you wanted to add something?
Just very briefly to point Millicent to the fact that in the past year we've really done a lot of work to enhance our policies that are meant to support some of the issues you are addressing, you're raising around, supporting women, for example. So the whole issue around diversity, inclusion, safeguarding, policies around bullying and harassment, issues around maternity, paternity, adoption, and sick leave. And I just want to point you to the ladies not seated not too far from you. Her name is Susan. She's a senior grants officer and responsible for the DELTAS and also advising on policy. So she can tell you a little bit more about all our policies in that area.
Shweta?
Okay, thank you very much for that question. I think India Alliance was established essentially to capacitate research in India. So even if it is an African fellow or an African scientist, I'm not even touching it. Okay, so even if it's an African fellow or a scientist, they're welcome to apply, but there are certain criteria and conditions. Two of them, very briefly, I'm telling you, the details can be checked out. Sorry?
Maybe try that one.
I think it's... Okay, yeah, I don't know. I'm too electric right now, maybe. So there is an option called the work outside the host institution. So usually in India Alliance, the model is that you have to have a host institution in India which is ready to get you there and sort of support you during your research time. But there's also an option of work outside your host environment. So if you are based in Africa, you can do your major part here also, but it has to be an India-centric research question. So your writing or your analysis can be back home in your country, but you can. So there are different models of partnership between different countries that we are trying to look forward to. I don't know currently if we have any African student as a fellow, but we do have partnerships and mentors. So for me, for example, there are advisors on my team, for which I have chosen somebody from Africa, somebody from America, things like those. So they also give you an opportunity to collaborate via India Alliance. Thank you.
Okay. By the power in me vested as chair of this particular session, I end the question and answer session, and I have a few announcements before we applaud our guests and break for lunch. The announcements. The fellow named Somefu Dolapo Oluwaseyi has been included in session 3.2 this afternoon. Also, Diana Mania has been included in session 3.1. Pay attention very carefully. Session 3.1, which is Major Communicable Diseases 3, has been moved to this particular venue, this amphitheater. Session 3.2, which is Communicable Diseases 4, has been moved to room C001. C001. And Session 3.3, Neglected Tropical Diseases and One Health 3, has been moved to room B001. And of course, as Olivier already mentioned, Session 3.4, which I expect a lot of you to attend and have fun with Olivier, is moved to Session C005. Also, Session 3.1 will now be chaired by Professor Bashiru Bonfo. I think those are the announcements, and we can now applaud our speakers. Okay, thank you very much. Let's break for lunch, and then we come. It's about 13:40 right now. So plus let's try to get back. Not here. Let's try to get to our various parallel sessions at 2:30. Thank you very much.
the table on the right, we realize that there's been improved uptake of the antenatal care interventions amongst the pregnant women. For instance, if we take ITN use, it's improved from 3% in 2003 to 43% in 2014. And then looking at iron supplementation, it's improved from 40% in 2003 to 59% in 2014, according to the Ghana Demographic Health Survey reports. And then there's also evidence of decreasing malaria prevalence in Ghana. So we were expecting that in the face of increasing uptake of interventions to control malaria in pregnancy and decreasing malaria prevalence, there should be a commensurate decrease in the proportions of low birth weight and maternal anemia. But the graph on the right is showing us that in the blue line, since 2010 to 2017, low birth weight incidence has been around 11%, and there has been virtually no change in the country, as well as maternal anemia. It's also remained the same around 36 to 40, around 36 to, percent at term pregnancy. So we asked ourselves some few questions. What could be the likely reasons? We are thinking that probably we do not understand all the co-contributing morbidities to maternal anemia and low birth weight. And so probably that is why placing emphasis on only malaria is not helping us to solve the problem. There could also be unfavorable sociocultural context within which the women live, such that it's impeding the way they use these interventions. Or probably we are reporting something that we are not doing. It could be that there's suboptimal implementation at the antenatal clinics. For me, I'll be focusing more on the comorbidities probably being present and we not identifying them. But the rationale for this study actually is that the control programs have always or mostly depended on the proximal coverage measures. That is the proportion of women being reached with the various interventions that are being delivered at the antenatal clinics, and have not really looked at the impact. Although we are measuring or taking measurements and recording HBs at 36 weeks and low birth weight incidences, we are not really marrying what is happening to malaria with these outcomes. So this study was set out to determine the relative contribution of malaria to the persistent burden of low birth weight and maternal anemia in the context of decreasing malaria transmission in Ghana. Specifically, we want to determine the prevalence of malaria during pregnancy and other parasitic infections that we know also contribute to anemia and low birth weight, like intestinal helminths, schistosomiasis, and other infections, HIV, syphilis, all these STIs that could also be contributing. And to measure the proportions of maternal anemia and the incidence of low birth weight and other adverse pregnancy outcomes among the study population, we would determine the proportion of low birth weight and maternal anemia, what proportions of these are actually attributable to malaria, and then the infections, and then also determine some individual level factors like the socio-demographics, nutrition, adherence to interventions that could independently be related to low birth weight and maternal anemia in the study population. So this study is being conducted in 2 regions in Ghana, the Ashanti region in the middle, and then the Volta region to my right. The pregnant women being in the population are those who are visiting the antenatal clinic for the first time, and then of all ages and parities, anyone who comes there the first time and is willing to participate in the study, we recruit into the study. And it's a prospective, non-interventional study being done in the antenatal care clinics. And what we do is that when they come in, we follow them up from the beginning from their first visit until they deliver. It's being done in the antenatal clinics in specific districts that have been selected in the Ashanti and Volta regions. And then we are looking at up to not less than 2,500 live births from these pregnant women to be able to help us to estimate the contribution of malaria to low birth weight, looking at a fraction of 10% or more. So as I said, the women are recruited at their first visit, and then at the first visit, we take their history, do the physical examinations, and then record all the interventions that they receive at the antenatal clinic, as well as the labs that are done for them. The study takes samples, that's 2 ml of venous blood, and then stool and urine samples at the first visit. And then we follow them up passively at each antenatal clinic and visit as they come in till they deliver. At each visit, taking 2 ml of blood for full blood counts measures for malaria parasitemia, and also we do filter paper blots and storing in amongst a specific number some serum in trizol. At each subsequent visit, depending on their gestation, we are also looking at their willingness to give us blood or not. Sometimes they come in and they say that they don't want to. So for each woman, we are looking at a minimum of 4 blood samples to be taken from them during their visits. When they are enrolled, 2 weeks after they have been enrolled, we visit them in the houses to look to measure their level of adherence to the antenatal interventions that they have received. So we look at their ITN use, we look at the number of pills of hematinics they received and how much is left, and we take records of all of these. At delivery, live births have their weights measured, and then any other pregnancy outcome is recorded. These are preliminary results. The study is still ongoing, and I'm going to talk about only baseline information that we have so far. There's a total of 3,057 women that have been recruited, and out of these, we have 954 deliveries. The mean age of the women is 27.2 years, and more than 55%, almost 55% of the women are nullips and primips. That's first and second time pregnancies. The highest level of education of basically more than 70% of them is up to the junior high secondary level, that is at the basic. And then the mean gestation at booking is 14.3 weeks. Looking at the time of reporting to antenatal clinic, almost 50% of them, these women have reported during the first trimester. But what we are realizing is that reporting in the first trimester does not mean that they receive the antenatal interventions that they need. These will be explained later on with subsequent results. Reported bed net ownership is 61%, of which only 46% slept under the bed nets before the night they came to the antenatal clinic. Parasitemia amongst them is 7.05%. And then those with syphilis infection, 3%, and HIV infection, 1.5%. The mean hemoglobin concentration is 10.8, and out of these women, 54.5% of them have hemoglobin levels less than 11 grams per GL. And we have also realized that 70% of these have microcytic hypochromic type of anemia. The mean birth weight of the deliveries that we've had so far is 3.05, and the proportion of low birth weight is 8.9%. Preterm deliveries, 4.5% amongst them. So at the end of the study, we are trying to find out how much good we are achieving with our control efforts for malaria in pregnancy. What are the effects of malaria on the low birth weight and maternal anemia? And if indeed the low prevalence of malaria is causing no harm or to that effect to the prevalence of anemia and incidence of low birth weights, then this study would help us to also find some factors that are fighting against the decrease in low birth weights and maternal anemia. Thank you.
I will ask you to maybe come and sit, and then we'll ask questions at the end. Can we do that? Please write down any questions that you may have, and we'll do it all at the end. Can we now have Nancy Nyakoe, who is going to be presenting on plasma cytokines and chemokine signatures during acute infection, but not convalescence, distinguish what? Between children with different levels of prior exposure to malaria. A long title.
Okay, thank you very much. I'm Nancy, a PhD fellow from WACPIP, and today I'll just be presenting some of the results I have from part of my PhD project, where we are interested in looking at immune changes in natural infection. And for today's presentation, I'll be focusing on cytokines and chemokine signatures during an acute infection that distinguish children with different exposure levels. Sorry. So malaria is still a problem globally. This is despite the individual strategic efforts to control and eliminate the disease. We still find that we have millions of cases annually and 100,000 of deaths, and this is partly because of the limitations that come with the strategies that have been put in place, that like drug resistance and insecticide resistance. And we know that to date the development of vaccine has been rather slow, and even with the current RTSS vaccine that was licensed, it's not completely effective because it's only up to 30 to 40% efficacy. Therefore, we sort of agree that for us to eliminate malaria globally for good, it's imperative for us to develop a highly effective vaccine. And vaccine development has had challenges, mostly because of the complex multi-stage life cycle of the parasite, which presents itself with a lot of genetic diversity. Hence, we have the parasite having various ways of evading the immune system, and also different multiple infection and different clone infecting people in endemic areas. And to date, we are not clearly able to define which immune mechanisms that can be used to actually evaluate our vaccine targets. And this is a challenge because the malaria disease, how it presents itself, we have a complex host immune response that consists of the humoral and cellular immune response. And for us to be able to make a headway, it will be important for us to identify functional protective immunity that comes with a naturally acquired infection. In malaria endemic areas, we know that due to the reduced incidences and severity, we see that people, individuals living in these areas develop acquired immunity. And this has been shown to be slow because it's mainly, you have to be exposed multiple times for you to develop the immunity. And the figure there sort of just represents the natural outcome of exposure to malaria and how immunity develops in endemic areas, where we have, when your infected for the first time, you tend to have severe diseases due to the high parastemia, but after multiple infections, because you're now developing immunity to the disease or the parasite, you're able to control the parastemia. However, we know that the immunity to the disease is not sterile, because individuals in these areas still develop parastemia. It's just that they don't develop the disease because they are able to control the outcome, the clinical manifestations. And research has shown that there is a complex interplay of antiparasitic immunity, which controls the level of parasite in the host, and anti-disease immunity that controls the natural acquired immunity. And this, from what Sam presented, this is sort of controlled by the endemicity or the transmission intensity in endemic areas, where we have due to different exposure levels, they therefore bring out a difference into how soon or how fast individuals are able to develop immunity. And taking advantage of this, we can easily be able to bring or identify immune effector mechanisms that control and regulate parasites in endemic areas. So one of the things we were interested in while looking at the cytokine production, because the cytokines are part of the immune system, and they're involved in every part of the immune system, especially when you have an infection at the blood stage, where the parasites are presented to the host for the first time. You sort of get the activation of leukocyte subset or white blood cells that activate various inflammatory pathways that produce the cytokines, and this also is produced by the other adaptive immune cells like the T cells. So these are an additional contribution to the natural acquired immunity, and looking at cytokine levels can help us develop an understanding on their contribution to anti-disease immunity. And also taking advantage of the variation in exposure can also help us clearly establish what the role of cytokine is in the different levels of immunity in children. We therefore sought to determine the plasma cytokine levels over the course of an acute infection in children with different exposure levels. And in this case, the different exposure level for our case was using children from different endemic areas. So we conducted a hospital-based longitudinal study where we collected samples when they were sick at day 0, followed at day 7 and day 21 from 2 different sites. So in Ghana, we have different endemic areas with different intensities of transmission, and we took advantage of this to collect from 2 sites, Kintampo, which is towards the north, and Accra, which is at the central. So Kintampo was our high transmission area, and Accra was the low transmission area. And this, we recruited children who presented at the hospital, 15 to 14 years, with clinical symptoms. We did a malaria rapid diagnostic test to screen them and confirm with microscopy. And for this study, we were able to collect 60 samples for each group, but we, however, did age-matched sorting, and eventually we analyzed 40 samples from each group for the cytokine analysis. So we picked plasma samples and did a Luminex magnetic bead array assay to determine the cytokine levels. And we measured 25 cytokines, and this is just the list of the cytokines we measured. And all the cytokines, or some of the cytokines that were below detection limits, were excluded from any further analysis. And in our case, we had about 4 cytokines that were eliminated from further analysis. So for us, because our aim was to understand what are the differences in this cytokine production in the 2 sites and over the course of an infection, we did a comparative analysis for each of the cytokines that were significantly different, and we grouped them based on the function of the cytokines. And for the pro-inflammatory cytokines, which are mainly responsible for the clearing of the parasites, we found out of the 9 that were in our panel, we found 3 to be significantly different. The interferons, gamma and alpha, were sort of different, were highly significant during infection, and which went back to normal over time. However, interleukin 6 was significantly different in both sites. The chemokines also had a similar pattern, with some of them only being significantly different in one of the low transmission site, which was Accra, and the other 2 only having both sites significantly different. And the other group of cytokines which regulate the inflammatory response, we found 3 of them to be significantly different, which are shown here. And regardless of whether you are from a high or low endemic area, they all had the same, like during infection, you had high cytokine levels, which went back to normal after infection. So this just showed like both sites had similar ways of regulating the inflammatory response. We know that parasthemia is also one of the things that contribute to host stimulation or immune stimulation. And we are interested in looking at the correlation of parasthemia with the cytokine levels. And we found about 5 cytokines were affected by parasite density. However, the other cytokines had a weak correlation and were not necessarily affected by parasthemia level. So production of different cytokines may mediate a network interaction, a complex network interaction. And we were also interested in looking at how each of the cytokines interact with one another. And from this, we were able to compare cytokine interactions from the 2 sites. And when we looked at the 2 sites, we were able to see a distinct signature that could be able to differentiate the 2 sites. And this is just a correlation of the network. The distance between the different cytokines, it just shows the correlation, and the stronger the correlation is the red lines, and they will be shorter, and the moderate correlation would be the blue lines, which are a bit longer. And when we looked at after infection, we saw that since cytokines had normalized, the pattern or the network also normalized. However, when we looked at the cytokine network on day 21, the high transmission site, Kintampo, had a slightly different network pattern. And when we scrutinized this further, we found that we had samples that had parasthemia at day 21. And when we removed them from the analysis, we saw that the cytokine network normalized. So this was able to show us like there is a distinct signature associated with when children had an acute infection and when they had an asymptomatic infection. And during asymptomatic infection, they were mostly associated with immunoregulatory cytokines. So in conclusion, we showed that generally cytokine levels are higher in low transmission area. These are induced or modulated by mostly pro-inflammatory cytokines and immunomodulatory cytokines. And we have certain cytokines that are affected by parasthemia. And we were able to show a signature that distinguishes the 2 sites, and it was mostly dominated by interferon gamma levels in Accra. And also a signature that demonstrates or differentiates sorry about that. Yeah, I know, I'm finishing up. So yeah, that differentiates the asymptomatic and acute infection. I'd just like to acknowledge my supervisors and all the funders and all the hospitals and research centers that help us with sample collection. Thank you.
Thank you very much, Nancy. Is Matilda here? Yes? Matilda will be presenting on social, cultural, economic, and individual factors influencing prevention and management of malaria infections among pregnant women in Ghanaian communities.
Okay. Yeah, so my name is Matilda, and I'm on the same project as Gifty, a MACAD project, and I work in University of Health and Allied Sciences. My collaborators are here with me. The title of my presentation is Social, Cultural, Economic, and Individual Factors Influencing Prevention and Management of Malaria Infections among Pregnant Women in Ghanaian Communities. Malaria in pregnancy is a debilitating disease, and governments in sub-Saharan Africa are making efforts to prevent and manage it. As part of efforts to achieve set targets of MDG 5 and SDG 3, the Ghanaian state has instituted free delivery services through the NHIS, which is the National Health Insurance Scheme, 2 decades ago. So malaria interventions have actually been incorporated into the National Health Insurance Scheme. And through the antenatal care, women receive free insecticide-treated bed nets. They also receive SP, which is used for intermittent preventive treatment of malaria in pregnancy, and they also are also taken through testing and treating malaria in pregnancy. Normally, some will do asymptomatic treatment, but when it's a pregnant woman, they have to do testing and treating, and so that is the difference. But despite these interventions that have been going on for 20 years now, we still have issues of malaria in pregnancy. And then despite such interventions, Ghana, like other sub-Saharan African countries, did not achieve the reset Abuja targets of 100% of pregnant women having access to IPTP and 100% use of LLINs by 2015. As Gifty indicated to you, we still have negative outcomes such as anemia in pregnancy, low birth weight, among others in Ghana. And so the questions we sought to understand or to seek in a community was to understand how and why, despite implementation of interventions, some women are not fully utilizing them and thus suffer from malaria during pregnancy. So this is what we sought to find out. And to carry out such a study in the community, we needed an approach that tended to study the culture of the people. So we used ethnography in carrying out the study. And this was a bigger study that we carved a focused ethnography out of. We realized that because of the intervention, some women get malaria, some don't get malaria. So our target was to look at those who get malaria. What are the environmental, economic, and social issues that really influence them getting malaria? So for this focused ethnography, we actually went in for women who had actually been clinically diagnosed of having malaria in pregnancy. And we did interviews with them, in-depth interviews, focus group discussions. We also had case studies as well and did observations in health facilities for a period of 1 year. The study was carried out in 2 regions, that's the Ashanti region and then the Volta region, which are indicated on the map of Ghana, which is to my right. And we carried out a study in 8 health facilities. 3 of them were faith-based facilities and 5 of them government-owned facilities. And the study was carried out in 12 communities. And our study population included the pregnant women who were infected, as well as the health workers who attended to them and community members. And the grounded theory approach was used for the analysis, and data was coded with in vivo software and triangulated and analyzed, and that is the results I'll be presenting to you in a short while. And all ethical procedures were followed. So as I said, we did a bigger study of 79 pregnant women, we had 55 conversations with 55 of them and 14 case studies. And then we also did a focused ethnography to understand why some came out with malaria in pregnancy, and these were really those who were clinically diagnosed of having malaria in pregnancy. And so I present to you the findings of the study. The results indicate that sociocultural and economic factors influence women or influence they getting malaria in pregnancy. And issues such as staying out late with family members. Some women stayed out late to converse with family members, to do other things, interact, or to supervise even children. And so they were exposed to malaria, mosquitoes for several hours before going to bed. Some of them also had to stay out at night to engage in economic activities like selling food stuff and other things, cooked food and other for people. And that also affected them. So they were exposed for several hours before going to bed. So by the time they even went to bed, even those who used mosquito nets, by the time they got to bed, they had had enough dose of mosquito bites, and for that matter, the bed nets were not really that effective. And also, another factor was the influence of social networks and family members. Some of them were highly influenced by social networks. For instance, women who stayed in houses that most people were not using bed nets tended not to use bed nets. And then, or when family members were not using them, they were not really interested in using bed nets, and even issues of visiting hospital to benefit from SP and treatment, testing and treating. Some of them, while the social networks, their friends told them that, oh, it was expensive to go to a hospital. They didn't go to a hospital to access the free treatment or the free SP that is normally given to women after quickening, and that is 4 months during pregnancy. So this affected their ability to access the intervention. Also, gender power relations in households was also key, where women, some went to the facility, got the necessary education, got SP, but when they went home with a bed net, within the bedroom, husband decided whether he should use bed net or not. Where a husband felt that he didn't want a bed net, they had to share the bed without using a bed net. And women didn't want to oppose their husbands because they felt that that could lead him out to other women. And this was particularly common in the Volta region, where we have a strong patrilineal system. And so men have power and control. But in the Ashanti region, which is matrilineal, power balance between husbands and wives was quite low, and for that matter, there was not much influence in that region. Also, the inability to afford the cost of maternal health care and malaria and pregnancy prevention and treatment affected women's ability to protect themselves. We realized that though some receive free malaria and pregnancy health services, but maternal health care in general, they found it expensive because some of them had to do other testing and all that, and that was expensive for them, so some didn't even access the service. And also, so this affected some. They didn't even want to visit the hospital. Some also skipped their scheduled visits, and for that matter, they were exposed. And then also environmental factors. Realize that in Ghana, we have the dry and wet season, and the dry season can be very warm, degrees of up to 35 or 40. And so some of them were not comfortable during the warm season to use bed nets. And what even compounded the issue during the warm season was that some of the design of some of the houses was such that you couldn't even use bed nets during the warm season because it became really warm. Some of the case studies we visited at home, we couldn't even stay in the bedrooms because they were just too warm for us. So you can imagine what they felt even at night. Then individual attitudes to realize that some of the women got education all right, got the bed net all right, got home and didn't use it, or some of them even didn't refuse to take the SP at the facility, you know. And also some also trusted in other methods like the use of fan, coil, and spray, which when they used, they felt that it was enough, it was protective enough. And so they came down with malaria. I'll just read to you how these factors sometimes are interconnected, such that a woman encounters several of them, which results in she getting malaria. So this is a case of a woman who had come down with malaria. So she says, the interviewer asked her, how long do you stay outside during the night? I sometimes stay outside till 9 p.m. Do you have mosquitoes in the area that you live in? Please, yes. When was the last time you slept under the mosquito net? Almost 4 months ago. Why are you refusing to sleep under the mosquito net? I always switch on the fan when I'm about to sleep. I don't sleep under the mosquito net when the weather is warm. Who encourages you to sleep under the bed net? The man who impregnated me encourages me to sleep under the net. Do other members in your household use bed net No. What have you been using to prevent mosquito bites at home? Mosquito coil and mosquito net. This was an 8 months pregnant woman. She was 8 months pregnant and clinically diagnosed of malaria. So you see how the factors interconnect. This was a case of a woman who stays outside up to 9 p.m. She wasn't using bed nets. And then even the advice she got from the man who impregnated her, she wasn't even using the advice because her people within her house were not using the nets. So this illustrates how the various interconnection factors within the community influence how women access malaria and pregnancy services. So in conclusion, social-cultural, economic, environmental, and individual factors influence pregnant women's utilization of malaria in pregnancy interventions. Some pregnant women continue to remain vulnerable to malaria attacks because if they go to a facility, they get all the education, they get the resources, they go home, and within the social-cultural environment, they go back to the same attitudes and practices, which does not sometimes promote the use of the various interventions measures, so they come down with malaria, which subsequently leads to other undesirable consequences. So health service delivery needs to be strengthened in communities to encourage all pregnant women to access ANC interventions early and consistently. I'd like to thank my collaborators who are here with me, as well as my mentor, who's been very supportive, and also to my sponsors and to the communities that I worked in. Thank you very much.
We'll ask questions right at the end. Is Amadou here? No, so Amadou is not here. So we will move on to Martin Mbonye, who will talk about female role in their male partners' uptake of HIV services, experiences of male intimate partners of female sex workers in Kampala, Uganda. He's already.
Good afternoon, thank you very much. Switch gears a little bit, slightly from malaria to something else. This is one of the themes that is coming out of my ethnographic study. It focuses on the role of female sex work partners in encouraging their male partners to uptake HIV services in Kampala. So this is a brief profile of the HIV situation in the country. Most of the new infections tend to happen among key populations, and female sex workers are part of that, the key population. About 5 times more HIV infections among this population compared to women in the general population. The same is true for the intimate partners of female sex workers. And intimate partners are men who are in non-commercial sexual relationships. Sometimes they stay together, sometimes they have children together. So these men tend to have a lot higher HIV prevalence rates than other men. However, these men are rarely a focus of attention in interventions, and yet we need to get as many people onto the into HIV services as much as possible. So how can these men be helped to access services? This is the progress of Uganda towards the 1990 by 2018. In the picture there is the Director General of the Uganda AIDS Commission. Recently he said that actually the situation has improved. There are about 90% of all people diagnosed with HIV are receiving sustained antiretroviral therapy, and all people receiving antiretroviral therapy have viral suppression. This is quite impressive. However, there's a catch. He says there are only 55% of men and boys living with HIV know their status. And so this is where my focus is for my PhD. My PhD focuses on masculinity and its influence on the uptake of HIV services among intimate male partners of female sex workers. For this particular presentation, I'm focusing on objective number 4, which is assessing how female sex workers make sense of their relationships with their intimate male sexual partners and what influence they have in their sexual partners' uptake of HIV services. This is an ongoing study. I started ethnographic data collection in January of this year, almost halfway there. In the red there are the people who I have interviewed. I'm conducting in-depth interviews as well as focus group discussions. And of course, being an ethnographic study, my main method of data collection is participant observation. And I've done this with all the participants that have been recruited. There are 2 main categories of men, just to explain that a little bit. So both of them are intimate partners of female sex workers. I get these men from NMRC cohort, which has been offering services to female sex workers since 2008, and it opened up space to their partners around 2011. However, of the about 3,000 women, only 250 male partners of these sex workers have managed to come and attend services. The others have refused to take up services or have given one excuse after the other. In my ethnographic study, I am contacting both categories of the men. This study is conducted in Kampala, and that is a map of Kampala. It's a multi-ethnographic site, a multi-sited ethnographic study, which means it's not, I'm not based in one site. Once I recruit a male partner, we have a conversation, and he allows me into his space. It could be his home, it could be his workplace, it could be his place of leisure. And the pictures show the various places that I've been visiting. Now, I would like to share with you how female partners assist their men by using three experiences of 3 men. These are not their real names. The first man is called John. John is a 40-year-old border rider. Border rider is like an Okada rider for the Nigerians. If you see in the upper picture on the right, those are very common sight on the streets of Kampala. They follow all the traffic rules, as you might imagine. This gentleman lives in a single room. I was very fortunate to be invited into this man's household. This is something I don't take for granted. He stays with 5 children. It's only one of those children is his, according to him. The rest is not sure. Now, he's supposed to be the main breadwinner, but in many cases, he doesn't manage to fulfill this role of breadwinner. This is something that makes him a little bit uncomfortable. However, his history of HIV was one where he struggled a lot to come to terms with it because he felt it was damaging his masculinity. It took him so long to access services until he met his female sex work partner who encouraged him, and after a period of reluctance, he managed to go, and he was put on ART. Interestingly, the wife is HIV negative, but she continues to live with him. This is the only couple where the man lives with his female sex work partner, and they seem to be going well. Interestingly, after getting better, after recovering, after starting ART, he resumed his some of those masculine norms which he was known for in the past, and he has a lot of concurrent sexual partners. He's quite proud of this. The next person I've had a pleasure to interact with is Peter, who is a 35-year-old primary school teacher. He has 2 children. He prefers to identify himself as a born-again Pentecostal Christian. Peter came from another district and settled in Kampala. He suspected that he had HIV, but he couldn't quite get himself to seek treatment. He had seen one of his uncles succumb to a lot of stigma when he disclosed his HIV status. So he just stayed without any help until he got this female sex worker partner. Because she was attending the clinic, she encouraged him to go and access the clinic. And after a while, he agreed and he started ART. Lastly, this is slightly different. James is a 45-year-old university graduate. For a year, after meeting his sex work partner, the sex work partner went and tested and found that she was HIV positive, and she blamed him for the infection. So he lived for a year assuming and suspecting that he was actually HIV positive. This included high-risk behaviors, alcohol, and so on, until he picked courage, went to the clinic, and he was pleasantly surprised to find that he was HIV negative. So it became confusing. This relationship is a very toxic relationship. They're up and down. His family does not encourage him to continue with the sex work partner, but he feels that being the mother of his children, he has no choice but to stick with her. He admits that he's at high risk of HIV infection because his partner is not adhering to ART, to the extent that they have been contemplating, together with the counselors at the clinic, to start him on PrEP. So it's something that he's thinking about. So in summary, from those three cases, we see that female sex workers are playing a very important role as messengers, as well as for those who are taking ART, the example that they show the men kind of comforts them and helps them to start on ART, even when they had feared a lot of things. Because in their peer male groups, they tend to exchange a lot of toxic information about what it will mean if they started ART and so on. However, the restoration of health sometimes also means the restoration of traditional forms of masculinity, and for some of them, like John, this means resuming the activities that actually led him to risk. So in conclusion, women are very legitimate link to treatment. The idea of the technical know-how is that they know the health system very well, so they have been helping their men to navigate this health system. But men continue to struggle with traditional notions of masculinity that affect their health choices. Thank you very much. Don't take the participants for granted, because an ethnographic study is very invasive. You enter people's spaces. I've been allowed to enter their homes. I've formed acquaintances, so I'm very grateful for the participants, and this study continues.
To Koseyaza Dube. I don't have the title of your presentation here.
Thank you. I think maybe not having the title also shows that it's another shift from malaria to something different. My name is Koseyaza Dube from VETS, and I'm looking at informal social security and how it promotes human health. And I'm specifically looking at the stockfalls. A stockfall is a kind of, or a term that is uniquely South African that refers to informal social arrangements. If some of you know the rotating credit and associations, and also the accumulating credit associations. Most of these are quite common in South Africa, and quite a lot of women participate in these. I think the picture here that we see, it's a picture that might be common in so many different African societies, where women are kind of the most who do the informal trading, and at the end of the day, because this is a laborious work that they have to do, and it requires so much attention, and also it requires so much of their time as well. So most of them spend their time in these settings, and at the end of the day, most of them also don't get the opportunity to visit or to attend or to go to any healthcare facility for any other ailments that they might be having. So Having looked at this, I think also when it comes to social security, most of the population within the world are excluded from any form of social security. And worldwide, it is estimated that a total of 5 to 10% of the working population is covered, and that also exclude quite a number of people. And in South Africa, the exclusion is even more pronounced because of the triple burden of that we have of unemployment, poverty, and also the issue of inequality. And this exclusion forces people to rely on informal social security as means of social protection. In this place, I use social protection in 3 different forms, as it has these 3 different functions. The first one is to address the income insecurity, and the second one, which is my main, to enhance access to education, health, and other social services, and also finally to strengthen livelihoods. In South Africa, when you look at the issue of stokvels, these constitute an important part of the informal social security system and have become one of the indigenous mechanisms used to increase savings and sustain livelihoods and improve social and economic well-being for the poor and for the vulnerable groups. However, there seems to be a gap on studies that focus on the role of informal social security in meeting the needs of the poor, and this includes their health needs. So this was the focus of my PhD. And the framework that guided the study was the sustainable livelihood approach, and with the aim of exploring the contributions of stokvels toward meeting the social protection needs of their members, and with a research question that focused on how these stokvels meet the social protection needs of their members. And this was a qualitative study, used a multi-case study as a design, with focus group discussions and also individual interviews being used as methods of data collection, and this data was analyzed thematically. And in the findings, it kind of emerged that yes, stokvels do contribute to income insecurity, and they also contributed to the educational needs of their members, and it also enhanced access to a broader social capital base. And at the end of the day, it also kind of strengthened the livelihoods of its members. However, what was quite interesting within my study when I was doing this, what I observed within these women is that the other thing that these stokvels did was to promote indigenous knowledge in dealing with health-related issues. So because of the easy access to indigenous forms of dealing with health and illness and also with well-being, most women kind of encourage each other to kind of follow that route. And in some cases, they even shared what one might have got from a traditional healer to deal with a particular ailment with a colleague who might be presenting with similar issues or symptoms. And at the end of the day, I just thought about this, that is the information being shared the correct information, or there is a need for maybe some form of intervention. And this kind of gathering promoted health information sharing amongst these women. It is in discussions like this that they don't normally focus only on their financial beings, their social capital, but it's also sharing of health-related information. But the challenge then was also the fact that people are sharing. It's also people without the expertise, and it's people that also don't have all the technical know-how. It's they are sharing because of their experience of a particular medicine or a particular intervention that they might have had. So when I did this study, the aim was to kind of contribute to guidelines that will inform policy development, especially for programmatic interventions regarding the recognition of stokvels as a form of social protection for the poor, and also the enhancements of knowledge within the field of social development of stokvels and social protection, but most importantly also to initiate debates in terms of formal social security networks and health promotion and or education. And to do this, to push this further, I just got the a Qatar re-entry grant, and where I'm going to be looking at how these platforms can be used in terms of hypertension screening among women in Soweto. There were some limitations like a qualitative study, issues around sample size versus the generalizability of the findings, and also sometimes socially desirable answers or responses from the participants. This was also, I think for me, when I started the data collection process, the first question that they asked me is that how much are we going to be compensated for our time, and are we going to have lunch if we are to attend these focus group discussions with you? Then the answer was no, because I'll be coming to your settings. And I asked, why are they asking that? And it's like most health related research kind of compensate people for their transport, and also they're given lunch for participating, and which is, I think for some of them, they've used that as a method of earning some income as well. So it then poses a risk of what kind of answers then do you get if somebody just comes because of that kind of motivation. So I am proposing a study that will use a mixed method approach, which will be both qualitative and quantitative, in order to explore this further, because I feel that at the end of the day, as much as we can have all these other interventions, at some point there is a need that we also think about the poor, those who are laboring, and I also maybe think of this might not be only a South African problem, but it might be issues of concern for different African countries where we have the issues of poverty and marginalization still very much pronounced within our community. So we might have to think about innovative ways of dealing with health and illness in future. I thank you.
Good afternoon, everybody. Let's come back to malaria. The topic of my talk is about utilizing spatio-temporal dynamics of malaria to inform and optimize control intervention. The presentation will follow the line out here. As you know, there is a decline in malaria incidence following the implementation of control strategy like the use of treated bed net, the intermediate preventive treatment with sulfadoxine pyrimethamine, and the use of artemisinin combination therapy. Both strategies contributed to reduce the incidence of malaria. In parallel of that, we observed that when the transmission decreased, malaria is becoming more and more focal. And the transmission is sustained by hotspot. Hotspots mean but a concentration of house block where we found more cases of malaria. In Bayangara in Mali, despite the reduction of the incidence of malaria report in many settings, we realized that the incidence was stable from 2009 to 2013 when we compared the result of the study to a study performed on 1999. We asked ourselves why the incidence is quite stable despite the implementation of control strategies. And likely in 2016 in Mali, the seasonal malaria prevention strategies have been scaled up. And again, likely in Bajangara in 2017, the indoor residual spread has been implemented in the Elf district. Let me tell you that Bayangara is a sentinel site of our National Malaria Control Program. It's also a field site for the research, Malaria Research and Training Center. That's why we are most focused on this site. in view of the implementation of new strategies and in view of the place of Bayangara, the role played by Bayangara in the national control strategy system, there was a need to update some epidemiological parameters of malaria as well to characterize the spatial and temporal distribution to provide evidence for targeting control intervention, mainly in our country where resources are very limited. The objective of our work was to determine the incidence rate of malaria, to measure the monthly prevalence of malaria infection and anemia, and to map and characterize the cluster of transmissions. Sorry. To do that, we realized a cohort study. The study was done in Banyangara, as I previously said. It took place from October 2017 to December 2018. We enrolled 300 children aged from 6 months to 15 years old, and we set two kinds of surveillance: the passive surveillance to catch the disease cases, and the active control, which consisted of monthly visit to catch malaria infection as well to measure the prevalence of anemia. At each visit, we perform a clinical exam. We collect blood for malaria smear, and we also measure the level of hemoglobin, and we collect dried blood spot. And all children have been georeferenced. We measure the latitude and longitude of the household where children live. And we use the software named StatCan to detect the clusters. As you see, the mean age of our cohort was 7.9, and here we have an age distribution of our participants. The female were dominant. The sex ratio was 1.54. And this figure showed the distribution of anemia, the prevalence of anemia and malaria infection. We realized that the malaria infection rate was 6.7, which was low when we compare to a period before the implementation of control strategies. And the anemia overall incidence was 2.4, which is also low when we compare to the period before the implementation of control strategies. And coming to the incidence, we realized that the incidence was much higher in older children than younger children. Before the implementation of control strategies, the incidence was much higher in children under 5 than the older children. This table showed that with the implementation of control strategies, there is a shift on the susceptibility of malaria. The older children are more vulnerable to malaria than the youngest children. Coming to the distribution, the spatial distribution of malaria cases, we found 3 hotspots. The first in the east part of the town, the second on the south, and the third hotspot were in the western part of the city. All of the hotspots were very close to the river named Nyame, which can be considered as a permanent mosquito breeding site. This figure, when we take in account this figure, and if we want to give some advice to our national control program, we may focus a particular attention to the point close to the permanent mosquito breeding site. As the keynote speaker said, that malaria is heterogeneous, even in local scale. Our national control program is not taking care of about the local distribution of malaria. The implementation of strategies cover all the region, irrespective of the hot spot or low spot. In conclusion, the incidence of symptomatic malaria was 0.5, which was much lower than the previous. And now we can say that the implementation of additional strategies, meaning seasonal malaria chemoprophylaxis strategies, and the implementation of indoor residual spray contributed to decrease malaria incidence. And subsequently, we realized that the old children are more vulnerable to malaria than the younger children, which suggests that we need to update our control strategies to expand a strategy like the SMC to older children. And also, knowing the marked spatial heterogeneity, we advocate for the target control interventions. And our take-home message will be to expand the SMC control strategies to all children, and also to have in mind the spatial distribution of malaria when we are implementing control strategies, and to focus more attention to the hotspots. I would like to thank here my advisor, mentors, and I'm grateful to Marcad, which allowed me to attend this meeting. Thank you for your attention.
Thank you, Risa. Thank you very much. So, Alphonsus, do we need to have one more person to present? No, yeah, all right. So we'll immediately proceed to questions. So the way we'll do it, because we're running quite late, and I know it's tea time, I would like at least each of our panelists to get a question, and I will ask Sam to give us a summary conclusion of this session. So questions, please. And let's try to make sure that each one of our speakers here gets a question. I found this quite informative. I've learned a lot about malaria personally this afternoon. So let's get some questions. Let me put it this way. I will not release you for tea until you ask questions.
Seconded.
So, hello. My question goes to Nancy, and my name is Daniel Kiboy. And maybe I missed this. And so, how was the response? I mean, how was the responses in terms of those who are? Do you check the asymptomatics?
Should I respond? Am I responding? Okay.
Yeah, maybe we can take a few questions before you respond. Is there anyone else?
I think there's one there.
There's one down there. Alphonsus behind you.
And Diana.
And behind.
Okay. Thank you for all the presentations. They were really great. My question goes to the ethnographer. You stated that in the course of your study you developed a very good relationship with your study participants, and you were invited into their spaces and so on, and you were able to really observe what was going on. Do you think that a female colleague of yours, like another PhD candidate, could do the same study that you're doing? If yes, why or why not? And then secondly, about the female sex worker partners, and let me talk about the third candidate that you have, the insurance broker. Is this a permanent partner that they live with in the house? And does the sex worker, is it a retired sex worker, or is she still continuing with the sex work that she does? And also this applies to all the others who seem to be living with the sex workers. And if so, it's a permanent partner that they're living with and they're continuing with their trade. What about condom use amongst the couples?
Hi, thank you. Thank you to the panel members for a really wonderful session. I thought it was really informative and a great range of topics. So thank you. My name is Julie Ballin. I'm from the University of Sheffield. It's an open question really, but perhaps mostly for the ethnographers, anthropologists and social scientists on the panel. In some circles, there might be the notion that social science research isn't as powerful or rigorous as the sort of more hard sciences research. And in terms of getting your results into, having impact from your studies and getting them to either change policy or practice, what would you say to someone who might critique the social sciences or qualitative research? How would you go about defending the type of study that you have done? Thank you.
All right, maybe let's have some responses.
Okay, just to answer your question, Daniel, when we designed the study, we didn't plan to look at asymptomatics. So the ones we came across during follow-up, because we are doing follow-up, those are the ones that were able to inform us about the response we are seeing in asymptomatics.
Thank you very much for those very interesting questions about the ethnographer. There are two of us, by the way, the ethnographers. But in terms of could a female person conduct the same study, in the literature on ethnography, and I've seen some PhDs which have actually been conducted by female persons on the topic of masculinity. The way I approach the homes of these people is I get permission from the female sex worker who is already registered at the MRC cohort, and then I get contact with the gentleman. We have a conversation before and agree on how we are going to be meeting. So I guess if you follow the same, you might be allowed into some spaces because of that very same question and sort of fear that you might have. The rapport building process can be tedious, and one has to take a lot of time to ensure that they are accepted in some spaces. So for some participants, it's easier for me to go to their homes, while for others, they would rather take me to some other, like a workplace, where they are more comfortable. In terms of the relationships between the women and the men, whether they are still engaged in sex work, yes, these women are in relationships with these men and they continue to do their sex work activities. I'll give you an example. The gentleman I talked about who is staying with his sex worker partner contacted her through a commercial arrangement. So he was a client first, and then she became a sex worker. Remember, a lot of these men are also enjoying material and financial benefits from the sex work partners. Of course, it creates tension in the home, but once you find the woman already engaged in sex work, it's very difficult without an alternative to get her out. For the insurance gentleman, he doesn't stay with his sex work partner, but every once in a while they meet up. And the reason why he doesn't stay with her is he is staying with his other relatives, other men. So they just make arrangements somewhere and engage in relationship. That relationship has got a lot of instability, but that's the nature of relationships anyway that they have. Condom use is very, very difficult in these relationships. Even for those in discordant relationships, it doesn't last. It's very inconsistent. And I think it's typical of people in a sort of marital relationship. How do you engage in consistent condom use? Particularly if the men have not...
Can you please start concluding your response?
Yeah, I'm enjoying this answer, so maybe I'll stop there and we can continue over tea.
Okay, thank you very much for the question. Well, for the relevance of anthropology or social science to the case of, I'll use the case of malaria as an example. As we move towards controlling and eliminating malaria, we know that now it is beyond the point where you believe that when you just have particular interventions, people are rational and people will take, the uptake will be great. What we realize is that human beings, as we are influenced by various factors such as our culture, our norms, our environment, and all that. And so behavior is key to understanding and comprehending how human beings behave. And this is where social science meets the natural sciences. And you realize that when Gifty made her presentation, she presented figures and percentages and all that. And you realize that my presentation explains why and how we have these low figures. So to understand human behavior, you need to understand the norms and culture, and this is where we come in. So we tend to complement the natural sciences. So I think it's key in malaria research. Thank you.
Do we have any more responses from our panelists before I ask Sam to give us a... Yeah?
Okay, maybe just to add on the relevance of social sciences in terms of research, I think when I look at the current study that we're going to do, especially on having to explore the attitudes of people in terms of introducing a health component or a health session within their monthly meetings, then after that, then we can then look at the acceptability and uptake where hardcore sciences will also come in. People can screen and also refer. And then there's that early detection of especially hypertension, because it's coming up as one of the major killers now in South Africa. So we need to kind of work around that and the strategies that we can reach more people than those who can have time to go to the hospital. So we need to work around the clock and see how best we can do this.
Thank you. I'd now like to ask Sam to give us closing remarks.
Yeah. Okay, so this morning I think I was talking to Tom or to Alphonsus, and I was saying that I was surprised how little social science and health systems presentations there were. And although I come from, as you might guess, a very biomedical background, I have been very much adopted and drawn into social science and the power that it has to help us translate what we are seeing in the lab or developing eventually to be used as public health tools and interventions. So I think it's been quite a good session, and we have moved from basic sciences of malaria, trying to understand immunity to malaria and ways in which you can define immune status. We have also looked at mapping of malaria using spatiotemporal data and how that can inform interventions. And then we've moved on to ethnographic studies, which I think you've already discussed to an extent. And also a very interesting study around the use of what in East Africa we call them chama. And chama is the same thing, it's women come together, but surprisingly now, actually there are more male chamas probably than women. The men just looked at it, thought it was a great thing, and have taken it up and pushed it to the next level. And so the women have also had to move beyond just sort of small contribution to also make them very powerful organizations. So it's quite interesting to hear how that is being used in South Africa. So I think overall a really great session. And maybe just one final comment regarding this and the other sessions that we have shared, especially for the rapid talks. You may have felt that maybe the chairs are sometimes rather tough on you with time. I think it's absolutely important. Those of us who sit on interview panels for grants, or if you go to the Wellcome Trust for your next level of grant, if you go to MRC and all those, they tell you have 3 minutes. And they genuinely mean you have 3 minutes. If you try to go beyond 3 minutes, they stop you. So it's really important that you get to your message as early as possible. I did a calculation. For most of you, spend about 6 of the 10 minutes on the introduction, and particularly on introduction parts, which were often repeating what everybody else already have said. You know, malaria has 1 million cases and things like this. So it's quite important to get the message early so that by the time the time is running out, that's when the chair is harassing you to get on, and that's when you've come to the sweetest part of your message. So try and get the message as quickly as possible, and then that leaves you a bit of time and even the speakers to ask questions if necessary, the listeners to ask questions if necessary. Thank you.
So, with those words of wisdom, I think we can conclude and say to our panelists, to Sam and our fellows, thank you very much. I hope you all enjoyed it as well as I did. And we can break for tea.
Okay, just some quick announcements. We have a gala and awards night this evening at 6:30 at Salon Adriana. I think that's where we have been having lunch. So you are all invited. For the DELTAS fellows who have been here for the past 3 days and have not said a word or asked a question, you can be sure I won't be inviting you to the next DELTAS Africa meeting. If there are any program managers or program directors from the 11 DELTAS programs, I see we still have a lot of useful program documentation that was shared. I encourage you to take some along. You might just be a saver to somebody who would be interested in joining one of the programs. And for some of the materials that are left behind, I encourage the program managers to recover them because they are still very useful. And it costs quite some money to prepare them. So please, let's make sure that we don't leave them here. And then you are welcome to join tea and poster sessions, and then we meet for gala and drinks in the evening. Thank you very much, and you've been such a good sport, all of you. Thank you. Yeah.